We can list a to-do list on the board. Today is already Thursday. That's hard to believe. When we start Unit 4, we're going to go through first our review of nervous tissue. We're going to see neuron types. There's going to be 3 structural types. And we're going to see 3 functional types. Keep that consistent. So neuron types, and then we're going to see types of neuroglial cells. There's going to be 6 neuroglial cells total. We're going to compare the central nervous system, which is going to be the brain and the spinal cord. 4 of those neuroglial cells live there. And we'll talk about the peripheral nervous system, all of the neurons outside of the brain and spinal cord. 2 of those neuroglial cells live there. So if we can get through the first PowerPoint where we talk about the types of neurons, I'll feel good about that. The second PowerPoint, that neurophysiology PowerPoint, is talking about how neurons send signals to one another. And that's what I wanted to at least set up somewhat right here. So let's try this. I'm going to draw a type of neuron, and then later on when we get into the PowerPoint, you should be able to tell me structurally what type of neuron I'm drawing. And not a great artist here, but I'm going to draw something that looks like this. Put some root-like structures coming off the side. I'll put a little nucleus in the middle. All right, and I'm going to draw another one here. Let's put one. Just to give me some space, I'm going to make this one cut right across the middle here. I'll put one more short one here. Okay, not the best artist, but what I've attempted to draw here are 3 neurons. And when we look at a typical type of neuron, there are some parts on it that we need to label. We're going to call, well, this thing here with the nucleus in it is the cell body. There are these little root-like structures that we're going to call dendrites. And then there's this little spot right here that's kind of important. It's where the cell body tapers, it starts to narrow, and then there's this long cell process that we call an axon. That little thing, this little bit right here, where the cell body tapers and leads to the axon, this is an important structure. It's called the axon hillock. All right, and we already know this thing because we've seen this part of a neuron already. This is the axon terminal. And inside that axon terminal, like we've already seen, there are vessels full of those neurotransmitters. Let's label that too. A common neurotransmitter is acetylcholine, so I'll put it here as acetylcholine. We'll put those little dots of acetylcholine in there. Let's see if we want to add anything else. The anatomy is there. Okay, that's one thing. So we can see downstream we've got other dendrites. We can see once again the cell body. We can see this axon hillock that's important. We can see the axon. And then down here we have our other axon terminal. Anatomy is we're doing okay so far with the anatomy? because there's several things that I want to layer on top of this. We have to not only know the anatomy, but like where the physiology of this stuff takes place on the surface of the cell. And when I have to click away from a slide to show another one, it's hard to keep it in the head. So I'm trying to draw 3 connected neurons. And here's another layer that I want to add to that. Let me get a different color. When neurons are talking to one another, it's a lot like the stack of dominoes that you sometimes see, where there is a sequence in which these dominoes fall. There's a first domino that falls, then a second domino that's going to fall. Another way to say that is that these neurons are kind of like one-way streets. They don't send signals in 2 different directions. Dendrites are the ones that always receive a signal. Hello? You are just in time. No, you are fine. This really should feel like a party. Maybe I should start bringing snacks. I can't bring drinks in here, even though everybody's of age. But maybe I should get some better lights or something in here, some mood music or something. Really shouldn't be that stressful. But anyway, it gets fun here because we start talking about neurons. And this is like an everyday life stuff. So it becomes hopefully easier to burn into. So what I've got set up here is I've just drawn 3 neurons together, kind of like if you're setting dominoes up and one domino falls into the next domino. It's a sequence of events. And so this little green signal, that's my color that indicates electrical impulses. And this little electrical signal we're going to see is going to be picked up by the dendrites, and it's going to move through the cell body in one direction. So from the dendrites to the cell body, then to this axon, from the axon down to the axon terminal. And the electrical signal is going to stop right there. We said instead of the electrical signal jumping across this space, it's going to be a chemical signal. And in fact, right here is where it starts ringing bells from some of the stuff that we already talked about. We said when this electrical signal, when this action potential reaches the axon terminal, we already know on that axon terminal there are these little voltage gates for calcium. Let's remember those calcium ion voltage gates. They're voltage gates. So when this electrical signal hits those voltage gates, they open up. And just to the outside, I've got some videos we're going to check out of this too. When those voltage gates open, calcium is going to move into this axon terminal. It's going to surround those vessels of acetylcholine. And now with the calcium surrounding them, they can merge with this plasma membrane and release those neurotransmitters. I'll just open this one up, showing it release the neurotransmitters into that synaptic cleft. Now what we have here is a chemical signal. Right? Before this was an electrical signal. And now we've got this little space between 2 neurons where there's a chemical signal that's going to go from one neuron to the next. Yeah, no, good.
The signal opens up the gates, the calcium gates. Does calcium go through them, or the acetylcholine stuff?
So the first thing that happens is calcium ions are going to move into this axon terminal from the extracellular fluid. So calcium ions are outside the cell, out here in our ECF. And there's very little calcium ions in the cytoplasm of the cell. So there's this concentration buildup of a lot of calcium outside, very little calcium inside. And whenever calcium gates open up, it's going to flood into that cell very quickly and lead to the release of this acetylcholine. So 2 different things. Calcium comes in, acetylcholine goes out. Acetylcholine doesn't go out unless calcium comes in first. So now we've got acetylcholine making its way out. And before, we knew that acetylcholine had to land somewhere on this muscle cell where, let's get this one, had to land somewhere on this muscle cell where there were receptors for those chemical signals. On the smooth part of the muscle cell, there weren't any of those receptors for chemical signals. We said there were only voltage gates. So that's what I wanted to do is map on these neurons. This is the last thing that we map on there as we continue to talk about them. Yes?
Can you start over again one more time, please, from the dendrites, from the Reuptake of Signals?
I'll do it 3 times, and I'll start right here
Right.
on this one. So it's the same thing. We'll just see it 3 different times. So what I'm about to mention right here is exactly what happened up here on the dendritic end of this neuron. We said dendrites always receive a chemical. They receive some type of signal. And if we look at the dendrites and the cell body, I'm going to use a blue color to draw these little, I'll just draw them as little Y shapes. because I'm not a great artist, but these little Y-shaped structures, I'm going to put them all over the cell body. I'm going to put them all over the dendrites. And I was going to go, that's right where I was about to go back over here and do the same thing. I was going to put these receptors all over the cell body. And I'm going to try and use the same color. The blue color, these are going to be chemically gated channels. And I'll just write it over here.
So the chemically gated channels, that'd be like the calcium channels?
Well, this calcium channel is going to open and close based on a voltage. These chemically gated channels are going to open and close based on the presence of a chemical signal. Here comes this chemical signal. Right? These chemical signals, they can't just land anywhere. They have to land on a receptor that is just the right shape for that specific chemical. And that's what we were talking about with the muscles. Remember, this motor neuron isn't just touching anywhere on this muscle cell. It's going right on the little pocket of the muscle cell that has receptors for the neurotransmitters, has receptors for those chemical signals. And that's kind of what I'm trying to draw on this neuron. This neuron doesn't have receptors for chemical signals everywhere. Those receptors for chemical signals are only located on the dendrites and the cell bodies of neurons. Because as soon as we cross this axon hillock, we're going to see from the axon hillock all the way down to the axon terminal, this is where we're going to get voltage-gated channels. So there's voltage-gated channels for sodium. I tend to draw those in green. There are voltage-gated channels. I'll just say voltage-gated channels for potassium. And we also, we're just talking about the fact that there are also these voltage-gated channels for calcium. And if we want to map where those are, the voltage gates for calcium are always located on the axon terminals. So we got those there. They open and close based on a voltage. The sodium voltage gates, they start right here at the axon hillock. And they go all the way down to that axon terminal. So again, voltage gates are all along the axon. We'll do the same thing here, those voltage gates. That's what's important about that axon hillock. Here's where those voltage-gated channels start to get involved. And we'll see these potassium and sodium voltage gates kind of alternate with one another. So we don't see any of these voltage gates on the cell body or the dendrites. We said these voltage gates are like the dominoes, that if we can get one to fall, the next ones are all going to fall. And just to finish it off, let's make sure we have our receptors here. Put some vessels with neurotransmitters in there. Okay, now with that kind of set up,
So
what's up? What are you thinking?
Sodium and potassium both in the axon together, like.
That's right. Sodium and potassium voltage-gated channels are going to be anywhere between the axon hillock to the axon terminal. There's
So
an,
it's like kind of like a domino effect, like the first one receives the electrical signals and then it sends the chemical signals over to the other one?
that's exactly what I'm trying to do. I'm trying to show that there's an alternation between this what we're going to call. Maybe we can do it this way. And I'm going to get a little more detailed with my colors here. From the axon hillock, I'll try and use this color. We haven't used this one yet. From the axon hillock all the way to this axon terminal, where we are involving these voltage-gated channels. Electrical signals that involve voltage-gated channels we're going to call action potentials. So I think I tried to connect something last time, that action potentials and voltage-gated channels go together. So the next action potential wouldn't occur. Well, forgot I stretched that one way out. It would incorporate the entire part of the neuron that has voltage-gated channels. So the last action potential So kind of like what you were thinking, I think what you were trying to say there, that when we look at a chain of neurons communicating, you have these action potentials, and in between the action potentials, you have these signals that are sent using chemical signals and chemically gated channels. These, let me try and get a different color. I don't think that's different enough. We haven't used the red color yet. These places where there are chemically gated channels, maybe just between the cell, where we have a release of neurotransmitters, dendrites and cell bodies, these are going to be chemically gated channels, chemical signals. The electrical impulse is going to vary depending on how much chemical signals there are being sent. This type of varying electrical signal is called a graded potential. So over here we would have another graded potential. Even this one would be our very first type of, over here we didn't see where the signal came from, but this is a graded potential.
And the action potential starts at or after the cell payload.
That's right, because action potentials always involve these voltage-gated channels. And we're going to draw these voltage-gated channels. One of the things you hear all the time is that action potentials are always the same strength. And that's because the voltage-gated channels are going to behave the same on all of these neurons. They open and close at the same signals. So action potentials are going to be the same strength, like I said, always when we're talking about voltage-gated channels. But in these little spaces where we're talking about graded potentials, where there are dendrites and cell bodies and chemically gated channels, depending on the amount of that chemical signal, this is where I used that analogy of us standing on a dock and there's just smooth water around us, and we've got a sack of rocks, and we can throw tiny little pebbles into the water, and it just disturbs the surface of the water very faintly. Slight disturbances, that's analogous to just a few neurotransmitters being released, maybe a few neurotransmitters docked to these receptors. We saw these chemically gated channels in action when we were talking about that neuromuscular junction. I'm not, again, able to draw this accurately, but what we know is when an acetylcholine molecule docks to these receptors, we said that this was a non-specific ion channel. I can't spell. Non-specific ion channel, meaning that these chemically gated channels, they will let sodium in and potassium out, just like we saw in the motor end plate of a muscle. More sodium comes in than potassium goes out. So as long as these receptors are full of acetylcholine, that means that there is an influx of sodium ions. These little ions with a positive charge are moving into the cytoplasm of the cell. And that's going to make the cytoplasm of that cell become more positively charged. There's a term that we used that describes the cell becoming more positively charged. It's called depolarization. So we get a slight depolarization in the cell body. And again, you can think of that depolarization as the wave. We're throwing rocks into the surface of the water, and depending on how big of the rock, that's going to determine how much of a disturbance or how much sodium ion moves into the cell. So if we throw a big enough rock and the disturbance is big enough that enough sodium ions rush into this cell that they happen to trigger that axon hillock, again, the voltage-gated channels are like a series of dominoes that are set up. So as long as we get the first domino to fall, then the rest of them are all going to fall. So again, axon hillock is important because this is where we begin the action potential, where those voltage-gated channels are lined up and ready to fall. We will spend a good bit of this lecture talking also about the positions of these voltage-gated channels. How are they set up prior to getting pushed over? And when they are pushed over, how do they go through the series of positions to reset back to their original position? Thoughts? What are we thinking about?
What happened to the motor end plate?
So that's a great question. Motor end plate, anytime you hear motor end plate, we're thinking that is a structure on a skeletal muscle. That's the pit on the sarcolemma of the muscle that contains those receptors, that contains these chemically gated channels. And now that we're in Unit 4, we're no longer talking about the muscle at all. We're just talking about two neurons communicating. So one neuron, instead of talking to a muscle cell, it's going to be talking to another neuron. And when we look at the structures on these neurons, it's specifically not going to talk to the axon. It's not going to talk to the axon terminal. There's no places for those chemical signals to land there. It's only going to talk to the dendrites or the cell body, where those receptors for chemical signals are located. Just like the neuron's only going to talk to the motor end plate on the muscle cell, because that's the only place where there are those receptors. Good question.
You said it only talks to dendrites and what else?
Dendrites and cell bodies, because the dendrites and the cell bodies are the only place where you find those receptors for the chemical signals.
Are the dendrites usually in the same place and the terminals going toward the same direction, or the axon terminals usually are connected to the dendrites, or is it?
So you're thinking there are some different structural types of neurons. So every neuron is going to have a cell body, it's going to have dendrites, and it's going to have axons. But sometimes we'll see the cell body will be like off to the side on a stalk, and the dendrite runs directly into the axon. What I've drawn up here, we're going to see that all of these types of neurons where there are dendrites on one end, well, multiple dendrites attached to a cell body and one long axon. This is going to be what we call a multipolar neuron. In fact, since we just listed multipolar neuron, we might as well go ahead and jump into the slides here and cross off the other three types of neurons. All of these we're going to be calling multipolar neurons. Well, I'll just do it this way. I'll jump down here, and we will sketch these things out real quick. When we think about types of neurons, there is a neuron that looks something like this, where you've got a dendrite. This would be the dendritic end. It's kind of hard to tell it's a dendritic end, but it runs directly into an axon portion. I'll just put the axon terminal there. And then on a little stalk off to the side is where you find the cell body. So this type of neuron, where you have the dendrites on one end, the axon terminal on the other end, and then the cell body kind of off to the side on its own little stalk. This type of neuron is going to be called a unipolar neuron. My knee is hurting. Get over that. So unipolar neurons have a cell body off to one side. There's another type of neuron that looks like this. It's called a bipolar neuron. A bipolar neuron have these dendrites that run directly into a cell body. A cell body is right in the middle. And on the other side of the cell body, there's going to be an axon. It's a terrible axon terminal, but I'll leave it. These bipolar neurons with the cell body right in the middle, not on a stalk off to the side, are also a type of sensory neuron. I'll just label this axon. And here would be the dendrites. So the only one of these neurons that have multiple dendrites touching the cell body is this multipolar neuron that we drew. So structurally, we have bipolar neurons with the cell body in the middle between a dendrite and an axon. We have unipolar neurons whose cell body is off to one side. And we have these multipolar neurons who have multiple dendrites attached to that one cell body and one long axon. We also have three functional types. Let me list these three functional types. There we go. I'm going to just drag these 3 functional types of neurons over here. 3 functional types. The first one that we'll mention are going to be sensory neurons. Sensory neurons are either going to be unipolar or bipolar. Again, maybe some additional information. When we talk about sensory neurons, there's going to be general sensory neurons, which are located all over the body. They're things like touch receptors. So general sensory, when we talk about general senses, these are typically done by unipolar neurons. When we talk about special senses, so hearing, vision, smell, taste, all of those special senses are accomplished by these bipolar neurons. There are sensory neurons that sense something about the environment. Then there are going to be what we call processing neurons. These are called integration neurons. Integration neurons do the information processing. They interpret this sensory input. You know, is my hand on a burning stove? Is it slammed in a car door? If it is, then what is the proper response? Well, probably to pull my hand away. So once the information has been received and processed, and we know, okay, we're going to pull our hand away from this car door that it's slammed in, the neurons that actually accomplish the pulling the hand away from the car door are going to be the motor neurons. So we've got a whole list of motor neurons. When we get down into the motor neuron division, we're going to talk about one branch of the motor division that is under voluntary control. This somatic branch, it goes to skeletal muscles. When we were talking about muscles, we said skeletal muscles were the only ones that we are in voluntary control of. So if we want to wiggle our big toe, we can wiggle our big toe. We get to decide what's going on. But there's another branch of the motor division called the Autonomic Division. And this goes to the muscle groups that are involuntarily controlled, so cardiac muscle and smooth muscle. Those are controlled by this autopilot, or the autonomic branch of the motor division. Anyway, I feel like... Sure.
Is that also called the pseudo-unipolar?
Yes, so depending on whether or not this axon forks into one or two, it can be called pseudo-unipolar. So oftentimes a textbook are going to group together unipolar and pseudo-unipolar, both general sensory. The difference is whether or not the axon splices off to one or two axons.
What was the difference between sensory and integration?
Sensory and integration? So sensory is just sending signals back towards the brain and spinal cord. In fact, let's do it like this. I'm kind of getting ahead of another diagram that we're going to do. I'm going to try to squeeze it here on the board because I feel like leaving this up here is generating some good questions. But I said one of the things that we're going to do is separate the central nervous system from the peripheral nervous system. I'll try and answer your question there about sensory and integration neurons all in one. When we're thinking about the central nervous system, this is really just the brain and spinal cord. So brain and spinal cord. I'm going to draw just this circle that represents the brain and the spinal cord. The brain and the spinal cord is going to be nested inside the peripheral nervous system. So I'm going to take a slightly bigger... Give me 1 second. I'm going to give myself a little bit more room. Won't be a perfect circle, but let's kind of drag that out a little bit. Just to kind of give myself my room. So this second circle out here, this is going to be what we call the peripheral nervous system. These are all of the neurons outside of the brain and spinal cord. This may be too much, but when you hear terms like, since we have the neurons drawn up there, I'm going to go ahead and get this out of the way because hopefully it will be easier to understand here. When we're thinking about the peripheral nervous system, you're going to see some terms like ganglia, and then the terms nerves. You can see that. Ganglia and nerves. Ganglia, this is a term that you see on structures out in the peripheral nervous system, and it's going to describe a collection of neuron cell bodies. So collection of neuron cell bodies. We're thinking of just the cell bodies in the dendrites. These are in large groups out in the peripheral nervous system. They're called ganglia. Let me show you one right here. When we get into talking about the spinal cord stuff later, this is a model of some of the spinal nerves. And can you see these? These are spinal nerves, but occasionally there's that little swollen bump right there. This is going to be what we call the sympathetic chain ganglia, or paravertebral ganglia. When you hear the word ganglia, these little bumps, those are collections of neuron cell bodies out in the peripheral nervous system. Going to be kind of hard to write, but if we go into the brain and spinal cord, which we're going to get into brain models next time, inside the brain there is also there are neurons in the brain. The collection of neuron cell bodies in the central nervous system is going to be called a nuclei. So nuclei and tract. These two terms are used in the central nervous system to describe the collections of neurons. So collection of neuron cell bodies out in the peripheral nervous system is a ganglia. I'll put the same definition here, collection of neuron cell bodies. But now this collection of neuron cell bodies is in the central nervous system, so we would call that collection of neuron cell bodies a nuclei. If we're not instead of concerned about the collection of neuron cell bodies, if we're thinking about a bundle of axons, those bundles of axons are called nerves when they're out here in the peripheral nervous system. So bundles of axons out there in the peripheral nervous system are nerves, but those same bundles of axons in the central nervous system are going to be called tracts. Now that I've filled that up with plenty of space, let me see if I can answer Jose's question of sensory versus integration neurons. So out here in the peripheral nervous system, we're going to have sensory neurons. We know there's two types of sensory neurons, general sensory, special sensory. These are picking up whether you're getting your hand slammed in a car door or whatever. The thing about sensory neurons is that they're always sending signals back towards the brain and spinal cord. Neurons are always like one-way streets. So sensory neurons are always sending signals towards the brain and spinal cord. Integration neurons are always found in the central nervous system. They never touch the peripheral nervous system. So here's where we're going to find our integration neurons. I'll draw one of these neurons. Well, I'll just represent it with an arrow. So we've got these integration neurons in the central nervous system. Their job is to receive signals from the sensory neurons and decide, is this a stomach ache that I have? Am I stepping on a tack? Is my hand slammed in a window? What's going on here? And then once you figure out what's going on, it's going to be neurons here in the central nervous system. These are the motor neurons. They actually start in the central nervous system, but are going to reach out into the peripheral nervous system. And they're going to go down one of two pathways, like we said, somatic, which leads to skeletal muscles. You're in voluntary control of this. And then the other branch is autonomic, where we're going to lead to smooth muscle and cardiac muscle. That's the autopilot, the involuntary control of the muscles. Is that kind of what you're thinking about? Okay. A lot of good stuff up there. But to put anything else down, we'll probably have to erase something. All right, I'll leave that up for now. We'll pull down some slides and we'll click through those slides to see if now that we got this base layer down, if the slides make any more sense. And let me know if there's any thoughts or if you want me to clarify anything further. Sure.
So for under review, when we're talking about the unipolar, multipolar, and bipolar, there's like one part where it talks about them and then it talks about the dendrites and the axon, and it says choose between one or many.
Oh, yes. Right. So we're looking at page 2 on the neuron review sheet. And kind of how I have them sketched here, you can see neuron labeled with the letter A is our multipolar neuron. Neuron labeled B is our bipolar neuron. And neuron labeled C is our unipolar neuron. Then below that it says neuron A, this is our multipolar neuron. It is going to have many dendrites and one axon. Then neuron B, that's our bipolar neuron down here, it's going to have just one dendrite and one axon. One dendrite touches that cell body, one axon goes away from it.
You said neuron A has
Many dendrites and one axon. The other two, for both B and C, it's one dendrite, one axon. So neuron B here, the bipolar, one dendrite, one axon. And in a unipolar neuron, there's one dendrite that runs directly into an axon. The cell body is off to one side. So only neuron A would you use the word many. It has many dendrites and then one axon. Everything else has one axon and one dendrite. And if we're thinking about, how many axons, how many dendrites, we're thinking about the structural classes of these neurons. Is it unipolar? Is it bipolar? Is it multipolar? If we think about what direction it's sending those signals, either towards the central nervous system, it's going to be considered a sensory neuron. Sensory neurons are always going back towards the brain and spinal cord. Motor neurons are always going away from the brain and spinal cord. And interneurons are always stuck within the brain and spinal cord. They never leave. They're in there processing inputs given to them by the sensory neurons and communicating to motor neurons to carry out their response. Any other thoughts about anything as we ease into some of these PowerPoint slides? I really feel like that was a bulk of it. I mean, we're going to draw the electrical signals and the position of those voltage gates, but at least knowing now that voltage gates are not located everywhere on the axon, they're only located from the axon hillock to the axon terminal. That's where we're talking about action potentials and voltage gates. Those go hand in hand. Chemically gated channels go hand in hand with these graded potentials, and specifically we're talking about just the dendrites and cell bodies where this occurs. And I think that's what Colleen was saying, that there's this, when a chain of neurons communicates, there's this alternating action potential, then graded potential, then action potential, then graded potential. So voltage gates, then chemical gates, voltage gates, and then chemical gates. Okay. Here's our neuron review, and some of these things we've already done. We've compared central nervous system and peripheral nervous system. We just put that diagram up on the board. We've seen 3 types of neurons so far. Let me get through the 6 types of neuroglial cells. And then in the second PowerPoint, where we talk about neurophysiology, we draw those action potentials and graded potentials. As always, interrupt me if you are thinking anything, and we'll try and cover it. So we just put this up on the board. If we're thinking about the 3 functional classes of neurons, here are the 3 functional classes: sensory neurons, integration neurons, and motor neurons. It's interesting that the original studies done on the nervous tissue was just trying to map out the direction at which these cells were sending electrical current. Maybe I should back up and remind everybody, when we were talking about nervous tissue at the very beginning, when we went through our 4 types of tissue, we said nervous tissue is composed of either neurons or neuroglial cells. There are about 10 neuroglial cells for every 1 neuron. Neurons are much bigger than neuroglial cells. Neurons are the only ones that are capable of sending these electrical signals that we're about to graph. So neuroglial cells don't generate any electrical current, but they just do a number of things that help support these neurons as they send their electrical signals back and forth. So neurons and neuroglial cells. Of those neurons, like we said, the first effort in trying to figure out what these cells were doing is just to graph what direction they were sending these electrical impulses. So sensory neurons are always sending them back towards the brain and spinal cord. We said motor neurons are always going away from the brain and spinal cord, and integration neurons are locked within that brain and spinal cord and have to process information, decide what the proper responses would be. Central nervous system, we said, was the brain and spinal cord. Anything outside of the brain and spinal cord is our peripheral nervous system. I will add this to the diagram. If we look at the sensory division, there are two things we could add to that. So all of those things that it's just saying are basically right here in this one slide. Let me jump to this real quick, or I'll go slow. When we think about functional divisions, we'll talk about the sensory division first. And we said these are always sending signals back towards the brain and spinal cord. So one term that describes that direction of signal is going to be afferent signals. Afferent signals are always sensory. They're always going back towards the brain and spinal cord. And what I wanted to add to our diagram there is the difference between visceral sensory and somatic sensory. We're going to see that in my example of stepping on a tack or getting my hand slammed in a car door, this is somatic sensory. Somatic, think superficial parts of the body. Versus visceral, this is deep, like internal parts of the body. So if I have a stomach ache, I just ate something weird and my stomach is flopping around, oh, I'm feeling weird. That type of sensory signal is coming from your internal organs. So that's going to be visceral afferent fibers.
Kind of like the difference between like somatic, like outside pain, and visceral is inside pain?
Yes, you got it. Somatic, yeah, superficial. It also kind of has to do with skeletal muscle and smooth and cardiac muscle. But you're right, visceral, internal organs. Somatic is superficial, involves skeletal muscles.
So the visceral involve all the internal organs? I mean, the viscerals, I said they include all the internal organs?
That's right. So any of those internal organs, all of the sensory neurons that go there are going to be visceral sensory. So they convey all of those impulses from your internal organs. If there's some type of issue where they need to communicate to the brain and spinal cord, it's going to be these visceral afferent fibers.
Your appendix is rupturing and it's like really bothering you, that would be visceral.
Visceral, that's exactly right. Motor division, which is, I guess, what we see on the next slide, is when we're going away from the brain and spinal cord. I tried to map out that motor division as having two branches of the motor division. The branch that we're in voluntary control of, the somatic division, skeletal muscles. And then the other branch was the autonomic division. I called this the autopilot. And we're going to see that there are two different settings on that autopilot. This is actually Unit 5 information, so we won't really get into the details of it yet. But when we talk about how that autopilot works, it has two settings. Either fight or flight, which is sympathetic, ramps everything up, or what we call rest and digest, and that is parasympathetic signals, slow everything down. Anyway, we basically saw that because this is what I tried to graph. In blue, I have sensory neurons that are sending signals back towards the brain and spinal cord. So sensory, these afferent fibers are either coming from superficial structures like the skin. This is somatic sensory, or if it's a stomach ache or an appendix or something like that, that signal is being sent back to the brain and spinal cord by way of one of those visceral afferent fibers. Again, the brain and spinal cord contains all of those integration neurons. They never get to leave the brain and the spinal cord, but once they decide what the proper response is, they send that response out to the motor division. And here we can see that motor division, like I had mentioned, has two branches. There's the branch that we're in control of, the somatic branch that goes to skeletal muscles, and then that autonomic branch that we're going to investigate further in Unit 4 that we're not in voluntary control of, but has two different settings of this autopilot, sympathetic and parasympathetic.
Could you say the difference between those two again? Like
Yes. Sympathetic division is fight or flight, so we're getting chased by a grizzly bear. Our heart rate increases, blood is diverted to the skeletal muscles and away from the digestive system because it's no time to digest our food. We need to be able to get air to our lungs, get away from this grizzly bear or take down that grizzly bear, whatever we're going to do. You don't have enough blood to be in all of the organ systems all of the time. So depending on if you're in fight or flight, blood is going to be diverted away from the digestive system and towards skeletal muscles, like we said, so that you can get away or fight. And the opposite setting, this parasympathetic setting, is called rest and digest. So after you eat a big meal, blood is actually diverted towards the digestive system and away from skeletal muscles. You don't feel like working out or going for a big run right after Thanksgiving dinner. You have a strong parasympathetic signal being sent to blood vessels that divert blood, like we said, towards the digestive system and away from the skeletal muscles. Slows down heart rate, slows down respiratory rate, makes you want to read a book and fall asleep on the couch after eating a big meal.
So it's like the sympathetic, like sometimes when people get really scared, they throw up. Is that part of that, or is that
Nervousness, that is something that can happen, yeah. So sympathetic division, sweaty palms, all of that is part of the sympathetic nervous division. Right.
So you say, what is it, parasympathetic? It slows down heart rate and what else?
Slows down respiratory rate. Think of it as rest and digest. So you're just eating a big meal and you're watching a show on TV and you'll fall asleep soon. Now that we've talked about the three functional types of neurons, as we already have on the board, we should be able to recognize these three different structural types of neurons. The first one over here is our multipolar neuron because we can see multiple dendrites attached to that one cell body. What they're calling here as the trigger zone, that's the axon hillock. That's where those voltage gates really begin. And if we're going to trigger an action potential, it starts right there. We just have to knock over that first domino. Right? So back here on the dendrites, this is us standing on that dock, throwing those little pebbles into the water. And if we throw a big enough pebble, then that wave, that disturbance of electrical signals might hit that trigger zone and get that first domino to fall. Anyway, multipolar neuron. Here we have a bipolar neuron where there is a dendrite that goes directly into the cell body and an axon that goes away. Cell body's right in the middle. And then in our unipolar neuron, we can see a cell body off to one side, and this dendrite runs directly into the axon. Pseudo-unipolar neurons are the same way. They just have an additional branch in the axon portion. Okay, so we've got three structural types of neurons. Here's one thing that I want to add to it. Well, we should be able to answer some questions about these neurons on the models before we get out of here. But anytime you see a unipolar neuron or a bipolar neuron, you automatically know we're talking about sensory neurons. All sensory neurons are either unipolar or bipolar neurons. However, when we get into the other two types, integration neurons and motor neurons, we're going to see that all integration neurons and all of those motor neurons are multipolar neurons. These are the most common types of neurons. That's why I've drawn a chain of these multipolar neurons. Okay, so that's one thing. Multipolar, just by looking at that, we know it's a multipolar neuron, but we don't know its function. It could function as an integration neuron or it could function as a motor neuron. Those structures tell us sensory neuron. Good so far?
You said the multipolar neuron is
It's either integration or motor neuron.
but the bipolar and the unipolar is
Always sensory. And here's some practice with that. So this thing here, we're looking at a cell body. We've got a bunch of dendrites attached to that cell body. There's a long axon going away from it. We know this as a multipolar neuron. Next one is a bipolar neuron. We can see a cell body right in the middle. On one side there's a dendrite, on the other side there's an axon. Sandwiched in between is that cell body. So it indicates a bipolar neuron. We know that's a sensory neuron. And then here's our unipolar neuron with the cell body off to one side. You can see that dendrite runs directly into the axon. It bypasses that cell body altogether. These are also sensory neurons. They tend to be general sensory, like touch receptors. I was just talking about how bipolar neurons and multipolar neurons are always sensory. whereas the motor neurons and those integration neurons there are always going to be multipolar. Motor neuron, association neuron. Again, these two are always going to be structurally multipolar neurons. Okay, there was a question that I wanted to ask about these things, but I won't yet because we need to talk about these blue cells. We need to talk about these neuroglial cells. So we're about to talk about the 6 neuroglial cells. These are not neurons, not these big cells, but all of these tiny cells that are surrounding neurons and doing different things for these types of neurons. The first page of your neuron review class handout has a place to list these 6 different neuroglial cells, and then out to the right you'll have to list which ones are found in the central nervous system and then which 2 are found in the peripheral nervous system. Here we are with the list. So in the middle of that first page, the 6 neuroglial cells are right here: astrocytes, microglial cells, ependymal cells, oligodendrocytes. Those are the 4 of the central nervous system. And then the 2 found in the peripheral nervous system are going to be Schwann cells and satellite cells. Let's go through and list what each of those are responsible for. As before, I mentioned that neuroglial cells, they outnumber neurons 10 to 1. However, if we look at brain mass, they only account for about 50% of brain mass. Neurons count for the other 50% of the brain mass, even though they are outnumbered by those neuroglial cells. Neuroglial cells are just much smaller than neurons. The first neuroglial cell that we'll mention, this is a neuroglial cell of the central nervous system. It's called an astrocyte. It's the most common of those neuroglial cells. The most common, the most numerous. It has a couple of functions that we need to mention. We're going to see that neurons, they do not like to be under any kind of pressure at all. You know this if you've damaged an area. If there's any kind of swelling to an area and pressure is put against nerves, it's painful. Nerves don't like pressure at all. So what these astrocytes do is they basically have all of these cell processes that stick out. That's where they get these names, astrocyte. And all of those little cell processes basically build this scaffolding so that the neurons can move through the brain and are not under intense pressure. Has anybody ever seen the scaffolding that they put around outside of a building if they're trying to get up tall on the building and fix stuff? There was a time where I had to lay bricks and make mud and send it way up on those scaffoldings when I was 18, and it wasn't great, but it reminds me of the scaffolding, I guess, and these structures that you can put around the buildings to get stuff up and down the sides of the buildings. The astrocytes are kind of that same thing to neurons. They provide this space that neurons can grow through. Kind of, yeah. I mean, it's not like they directly touch the neurons, but the neurons, maybe here's another way. I've got this dog that she likes to grab a toy and then run underneath the bed where nobody can get to her. And she's protected in that little space. Sometimes she'll run underneath the tables, long tables where there's a bunch of chairs. She'll get underneath the chairs, the legs of the chairs, and nobody can get to her. I'm trying to get her to leave somewhere, she'll go under there and I can't get her. Kind of like a shield, yeah. So the legs of the chair there that she's kind of walking through and nobody can get to her, that's how these neurons grow in between the processes of those astrocytes. So the astrocytes kind of connect and the neurons kind of weave their way through them. But they don't touch the neurons. What they're touching right there are capillaries that run through blood vessels. Let me grab one of these brain models over here. See how some of these brain models, they just don't show any blood vessels there? And other brain models will show the fact that you've got these capillaries that run on top of the brain. These capillaries are obviously underneath the skull. They're right on top of the brain. We'll see the brain is full of neurons. Again, neurons don't like pressure. And one of the things that is problematic if you get into a car wreck or you see those football players get on the head-to-head collisions, this actually happened to my grandfather, fell off a ladder, had a stroke on a ladder, fell off a ladder, hit his head on the concrete, and blood vessels in his brain swole up. So that's the other function of the astrocyte, is to form this blood-brain barrier. You'll see some of those processes of these cells, they wrap around capillary beds to make sure that those capillary beds don't leak. And any damage to the brain can damage that blood-brain barrier and cause these capillaries to leak out. Then that puts pressure on neurons and it causes people to slip into comas. Anyway, so blood-brain barrier is something that's important. Astrocytes surround capillary beds to prevent them from leaking in the brain, and they also provide that space for neurons to grow through. Largest and most common in the brain. We'll move. Oh, here's a picture of an astrocyte, a little bit better picture. You can see where they do kind of reach out and provide this little support for some of the processes of neurons. They don't really wrap around the neurons. We're going to see they just kind of give them space and support them. There's another cell that does actually wrap around neurons. It's an oligodendrocyte. We're about to see that. But astrocytes, again, you can really see them forming that enclosure around the capillary beds and then really just kind of reaching out to give these neuron processes their support. Next cell that we mentioned, we just went from the largest and most common neuroglial cell to the smallest and least common. These are microglial cells. They're tiny. They have these little tiny cell processes. But what they actually do is they act as part of the immune system. They're the only cell in the central nervous system that has an immune function. So if there is any pathogen that gets into the central nervous system that doesn't belong, these microglial cells are the ones that have to sense it out and get rid of it. So they're like roaming macrophages of the central nervous system. And another interesting thing that I recently heard about these microglial cells, it's been discovered that your long-term memory, you know how long-term memory gets altered? It's the job of these microglial cells that will cleave off axon terminals. Like, without sharing all my own crazy stories, you know, your memory gets altered, I guess, as you have these memories for long periods of time. And one of the jobs of changing the ways that neurons connect in the brain are these microglial cells that will cleave off axon terminals and kind of rearrange the connections of neurons. Like this happens all the time. Here's one example. Like there are so many songs that we have to learn in this band playing music that if I don't play, like there are songs that we learned a few months ago that I've forgotten already because I don't play them a lot. And you know, if you think, how do you learn something and then forget it, you know, a few months later, these microglial cells are constantly going through your brain. When you sleep, one of the jobs of dreaming is going through the brain. These microglial cells are going through and deciding, is this a connection that we need to keep or is this a connection that we need to get rid of? And that's one of the ways that you get all these crazy dreams triggered, because these microglial cells are going over and flipping over these axon terminals and deciding, is this something that we need to keep? Is that on the exam or can I get rid of that?
Is that called pruning?
Pruning. That's one of the things that happens, pruning those axon terminals. Anyway, there's our microglial cell.
Are they the phagocytes because they attack the pathogens?
That's right. They act as an immune cell, like a roaming macrophage. Here's our third type of cell. It's just those cells that you see lining this. This is one of the ventricles of the body. Let me try that again. It's a ventricle of the brain or the spinal cord. Technically part of the body, but I could have been more specific there. We're going to see that these ventricles of the brain and spinal cord are going to be filled with fluid. This fluid is called cerebral spinal fluid, and that fluid is constantly being circulated by these cells, these ependymal cells. Now, again, I'm from a small town, so sometimes I hear people say ependymal cells, or you can say ependymal cells. I don't care how you pronounce it, as long as you spell it right on the test, you're fine. ependymal cells or ependymal cells. These are neuroglial cells found in the central nervous system that do not produce the cerebral spinal fluid. They just circulate it. They keep it moving. Here's another picture of these ependymal cells. They all have cilia on their apical surface, and that cilia beats in a uniform direction that creates a current of cerebral spinal fluid that moves through all of the ventricles. This cerebral spinal fluid, I know we're kind of jumping ahead to central nervous system stuff, but if, we just talked about how neurons don't like pressure. And if we look at the brain and spinal cord, we're going to see that it's encased in fluid, encased in the cerebral spinal fluid. And that cerebral spinal fluid, it really acts to increase the buoyancy of the brain and spinal cord. Imagine it like if we were to get injured and we had to go do some rehab. One of the things that people get into in rehabbing injuries or whatever it may be is to get them into a swimming pool, because once you're in the swimming pool, you don't weigh as much in the swimming pool. There's not as much pressure being put on some of those joints that you might be trying to rehab. And the same process happens in the brain. This cerebral spinal fluid allows the brain and spinal cord to float in a column. So it's not gravity isn't pulling down on it as hard. Those neurons aren't under as much pressure if they can float in this column of cerebral spinal fluid. So ependymal cells don't make this fluid. Like we said, they just keep it circulating through the ventricles.
So that might be kind of a gross analogy, but like the big slushy machines and they keep spinning. Is it kind of like that? Like they churn up the fluid to keep it moving?
Yeah, slushy is not gross. You're making me want a slushy now. But yes, that's a good analogy.
The body's gross.
Yeah, that's right. So those would be the little moving wheels that are turning or whatever, and keeping the slushy stuff. Maybe a pina colada or something like that. Those, yeah, there we go. That's better. All right.
In one place?
What's that?
Does it keep it in one place?
No, the fluid will go from one ventricle to the next. We'll see 4 different ventricles and then a central canal of the spinal cord. And we'll see the fluid constantly circulates through each of those pockets. And then even around the outside of the brain and spinal cord. So the whole brain and spinal cord are floating in this column of fluid. So just to jump to this one real quick, this doesn't show the whole thing, but these pockets that we see, these are going to be lateral ventricles. You can see this tail, that's a big lateral ventricle. Each ventricle is going to have a structure in it called a choroid plexus. This is on your lab list. Those little red structures, those are called choroid plexuses. And a choroid plexus is going to be a bed of capillaries that leak fluid out. And the fluid that leaks out of those choroid plexuses is cerebral spinal fluid. So cerebral spinal fluid is going to be coming out of those choroid plexuses. There's a choroid plexus in each ventricle, but what circulates that fluid, like we said, is the fact that each of these ventricles and even this central canal are lined by those ependymal cells. And they provide like we said, the slushy type movement to keep that cerebral spinal fluid going from whatever ventricle it was produced in. Fluid will go from these big lateral ventricles down to this 3rd ventricle, then through this cerebral aqueduct down to the 4th ventricle, and then from the 4th ventricle it leaves through these little apertures. There's a median and lateral aperture where that cerebral spinal fluid actually goes and surrounds the outside of the cerebellum and the outside of the cerebrum. And this is what allows the brain and spinal cord to float in a column of fluid, just like if we were to all go to the deep end of a swimming pool and just kind of tread water there. That's kind of what the brain and spinal cord are doing. They're kind of treading this cerebral spinal fluid.
Okay, but the spinal fluid is in between those cells, or the cells are keeping it there?
Let's go back. So,
providing a barrier.
yeah, so those ependymal cells, when we go back to that other picture, those ependymal cells are just we'll say just lining this central canal, for example. And so they keep that fluid in the middle of that central canal. They don't let the fluid doesn't leak through, it doesn't make its way back into those ependymal cells. Let's go back to the ependymal cell picture. No, you're great. It's great to think about it now, So this same picture that we were just looking at, all of the blue stuff, that fluid-filled cavity, those are the ventricles. So the fluid would have been produced by the choroid plexus, those capillary beds. So let's imagine there's a choroid plexus right over here that has leaked out the cerebral spinal fluid. So now we've got the fluid here. We just can't see where it came from. The job of these ependymal cells is to not let that fluid leak into the brain tissue, let's keep it in the hollow ventricle and just keep it moving from one ventricle to the next. All right. No problem. So that's 3 of those neuroglial cells of the central nervous system. The 4th and final one is called an oligodendrocyte. Oligo just means few, and this dendrocyte, the other part of this cell, it's describing that we've got one cell right here that is producing multiple sheets of myelin. So these, what look like flat pages that are rolled up like a scroll, this is a sheet of myelin. It's an extension of this one oligodendrocyte. So we can describe oligodendrocytes, these one cells, like we said, they're going to produce multiple sheets of myelin, and each of these sheets of myelin, you can see, are going to wrap around an axon, just this portion of a neuron. So that's the last of these cells in the central nervous system. I think I have a picture here that shows us a cross-section through the central nervous system. We've got these large cells here, the astrocytes. We can see them supporting neurons and forming that blood-brain barrier around the capillaries in the brain. Then we've got these microglial cells, which are kind of serving as part of the immune system, doing the axon terminal pruning. Then we've got those ependymal cells that we said are just circulating that cerebral spinal fluid. And the final one right here are those oligodendrocytes. One cell, you can see it's producing multiple sheets of myelin. So this one cell is touching 2 neurons. It's wrapping around 2 separate neuron axons with those multiple sheets of myelin. So again, this myelin is just going to be a little insulating material that is produced by these oligodendrocytes. There are 2 cells in the peripheral nervous system, and one of them also produces sheets of myelin. There it is right there. It's called a Schwann cell. So the blue cells that look like little tic tacs here, those, each one of those is an individual Schwann cell. It's kind of like these oligodendrocytes in the sense that it produces sheets of myelin. Unlike these oligodendrocytes, though, we can see a Schwann cell just produces one sheet of myelin, and it wraps around just that one little part of the axon, as opposed to these oligodendrocytes that produced multiple sheets of myelin. So multiple sheets of myelin, we know it's an oligodendrocyte. If it's just one sheet of myelin, we know it's a Schwann cell. The other difference is that these Schwann cells are out in the peripheral nervous system. Oligodendrocytes, again, are only in the central nervous system. So, okay, so we see the blue ones here, which are the Schwann cells. Let's think, just as a quick little review question, structurally, what type of neuron are we looking at here? This one on the screen. What is this structurally? It's a unipolar neuron. So we know it's unipolar. That automatically tells us that it's also a sensory neuron. And we've seen Schwann cells. Let's talk instead about the other thing, which are the satellite cells. Satellite cells are these pink or purplish cells that you see surrounding the cell body of these unipolar neurons. So we can describe the location of these satellite cells. They have something to do with the cell body of unipolar neurons. But it's not completely understood what they're doing yet around the cell body of those neurons. So if you go to current versions of A&P textbooks, they have found that satellite cells exist, but exactly what they're doing in there to help the cell body out is not completely understood. And that should be exciting to you because that tells you that there's lots of room for things that we're finding out and investigating. So if you are interested in research and want to go in one particular direction, that's an area of hot research right now, satellite cells. They've been discovered and they're being tested right now to see what they're doing. Again, we know they're part of the peripheral nervous system neuroglial cells in addition to these Schwann cells. So let's talk about the Schwann cell just a little bit. This is, we've zoomed into a picture of one of these Schwann cells, and we can see the nucleus of the Schwann cell. What is really the main part of this Schwann cell is the fact that it grows out this really long plasma membrane, and then it wraps that plasma membrane up multiple times around the axon. And you have a lab list where it wants you to know, let me look at the lab list specifically. It says it wants you to know the Schwann cell. Well, this whole structure here is a Schwann cell. In fact, this model, if we hold it up, we can see these Schwann cells that are here. And you can open up some of these Schwann cells to see that layer of myelin has been coiled around itself to make these multiple layers of myelin. If we're thinking about those multiple layers of myelin stacked on top of one another, this is what's called the myelin sheath. Again, multiple stacks of myelin. If we go to the very outside surface of the Schwann cell, the surface of the Schwann cell, that smooth outer plasma membrane of the Schwann cell, is called the neurolema. Remember when we called the smooth surface of one of these muscle cells the sarcolemma, muscle cell plasma membrane? The plasma membrane of a Schwann cell is the neurolema. So if we were to grab this model and point to the surface of that Schwann cell, that would be, the surface again is the neurolema. The whole thing is the Schwann cell. The multiple layers of coiled myelin is the myelin sheath. And this is what I was just trying to describe, the fact that when these Schwann cells initially grow, they start to elongate their plasma membrane, and this plasma membrane wraps multiple times around the axon and creates that myelin sheath. The very outer layer of that myelin sheath is the neurolema. This might work. Wait. And I'm going to hold on to that video for just a second. What it actually shows is the growing of one of these Schwann cells. I just wanted to show this picture real quick because it's what we've drawn on the board as one of our multipolar neurons. We can see a cell body that's got multiple dendrites, this one long axon. And I can use this picture to help with some of the parts of your neuron that you have to label. Because it's not exactly clear what some of these arrows are pointing to. But one of those arrows is pointing to the nucleus of a Schwann cell, while another one is pointing to this little gap, that space in between two Schwann cells that we're going to call the node of Ranvier. And there's still another arrow that is just pointing to, let's see, the myelin sheath of the Schwann cell. That's just pointing kind of incorrectly just to the surface of the Schwann cell. In addition to those, the nucleus of the Schwann cell, the nucleus of the cell body, and the node of Ranvier, you've got things like the cell body, the dendrites, and then as the axon comes all the way down to the end, kind of the collection of axon terminals is called a telodendria. So maybe my fingers could represent a bundle of axon terminals that we would call a telodendria. So that's the very end of this neuron that you have sketched on your class handout. This is just pointing out the fact that, you know, when we were talking, I like having these neurons up on the board the whole time. We're going to see that neurons, we said, are like one-way streets where signals are going to go from the cell body down to the axon terminal. And these, kind of the direction, all of these neurotransmitters that we're seeing down here in the axon terminal, those were actually manufactured here in the cell body. And they were sent down there to the axon terminal to just kind of build up and wait to be released. When these vessels are going from the cell body down to the axon terminal, this is what we called interograde movement. Mitochondria, like we said, vessels with neurotransmitters are all going from cell body to the axon terminal. We're going to see sometimes materials are actually picked up by the axon terminal and sent back to the cell body and recycled. This is just called reuptake of neurotransmitters. And when it goes back the opposite direction towards the cell body, this is what we call retrograde motion of either those organelles or recycled neurotransmitters. I feel like I had just 2 slides left there, so let me say something else with this slide. When we get into our brain dissection, I know we're still like a day away from getting into the brain stuff, but we're going to do cross sections through the brain, and we'll cut it up like a loaf of bread basically and flip through these different pages. And when we're looking at the cross section through the brain this way, you can see how the very edge of the cortical tissue of the brain is kind of a darker gray staining, as opposed to some of these areas of the brain that are a little bit lighter staining. Can everybody make out that distinction between what we call the white matter of the brain versus all of this gray matter? Now that we have all these neurons, any place where you see gray matter on the brain, gray matter, it could either be dendrites, cell bodies, or unmyelinated axons. Let me get my clicker to go back across a couple of things. This is an axon that it's myelinated. It's got Schwann cells. It's insulated by these layers of myelin. We're going to see that the more myelin layers are there, the quicker this electrical signal can be sent from one part of the neuron down to the axon terminal. Some neurons don't have any insulation whatsoever. So if I can go back to this picture, any place where we see gray matter, these are places where there are no sheets of myelin. So dendrites, cell bodies, or unmyelinated axons. Any place where we see this white matter, these are just going to be, if I can go back to this picture, white matter consists of just the myelinated axon portions. So you're not even, you wouldn't even have an entire neuron there. You would just have the axon portion of that neuron that is heavily layered in these myelin sheets. That's what is responsible for the white tissue there. And then look, as we go deeper into the brain, you can see these dark patches here. These are going to be what we call cerebral nuclei. And I know I've covered it up here, but nuclei in the brain and spinal cord are collections of cell bodies and dendrites. Cell bodies and dendrites are never myelinated. They don't ever have any insulating sheets of myelin on them. They're never going to stain as part of this white matter. They're always going to be gray matter. Again, axons can be gray matter too, as long as there's no insulating myelin on them. But if we see white matter, those are the myelinated axon portions of neurons.
Does that white matter, it doesn't particularly mean that it works faster or it works more than the gray matter because of the myelin sheets?
Well, we can say that the gray matter does the information processing.
Okay.
And then, once all the white matter is it's just this axon portion of the neuron that's sending a rapid signal. So it's going to involve voltage gates. It's going to involve bunches of layers of myelin to insulate that voltage signal. The next slide is what kind of takes us into our discussion of how two neurons talk to one another.
I have a question.
Yeah.
So when there's like brain damage, showing like an MRI, is that like the same like when it's like white? Does that mean because it's damaged?
I'm not sure how the scarring would show up on some of there. If you've got dead tissue in the brain for whatever reason, I don't know exactly how that dead tissue would compare, lighter or darker. If that's, yeah, that's a good question. I don't know. I wouldn't want to guess. I know one of the ways that they look at some of that stuff is by trying to see blood flow, and you can put things. You can analyze what activities of the brain are more active or less active in ways that aren't just showing the gray matter or the white matter. That might be a way to detect that, but it wouldn't. I don't know how to distinguish it between the gray or the white, any color ways. So what I've drawn up here on the board is kind of like what I was attempting this slide to represent, where we've got this chain of neurons, and one neuron has to talk to the next. How we describe these two neurons is, and we know this slide is actually, it looks like they're showing those neurons touch, but like we've done several times here, those neurons, they get really close together, but they don't actually touch. There's this little space in between there, that synapse. So the neuron that is sending the signal is going to be called the presynaptic neuron. Again, that little space between the two is the synapse. And this second neuron here that's receiving the signal, we're going to call this the postsynaptic neuron. So presynaptic neuron is talking to this postsynaptic neuron. And I think you have images of this on the kind of the last two images you have on your class handout today. You can see a little bit of a presynaptic neuron. That last little, we'll call this our presynaptic neuron, and it's communicating to our postsynaptic neuron specifically the dendrites or the cell body of our postsynaptic neuron. Right? The presynaptic neuron doesn't talk directly to the axon portion. It has to go through the dendrites and the cell body first. All right, let me see if I can get one thing going.
Development of myelin in the peripheral nervous system and the
It's a multipolar neuron. Okay, so we got that. Second question, do you know functionally what type of neuron it is? Can you tell functionally what type of neuron it is? So I think I heard, and it's 50-50. I heard either integration or motor neuron. We know motor neurons and integration neurons are both multipolar neurons, which that one is a multipolar neuron, but there's another clue there that will confirm for us whether we're talking about an integration neuron or a motor neuron. Can anybody see it? The first part of that is right. It is a motor neuron as opposed to an integration neuron. And how you know it's a motor neuron as opposed to an integration neuron? Schwann cells, that's right. Schwann cells, these are little sausage links that are always found in the peripheral nervous system. So if we're thinking about a multipolar neuron in the peripheral nervous system, it has to be a motor neuron. This is where we're going with some of the exam questions. So, no? Good, all right. Note cards will be all right. I mean, this is, if it's new, right, it's kind of hard to fit in your brain somewhere, but we get some practice with it. Let's see it in motion here.
The propagation of the action potential along a myelinated axon. This multimedia presentation will be most helpful if you already have a good understanding of the Schwann cell and the electrochemical process of the neuron, called the action potential. The Schwann cell forms a protective covering around the axon. Schwann cells start to develop in the embryo and continue to increase the wrapping around the axon through childhood. This development increases the thickness of the wrappings, which peaks in adolescence. This is why teenagers have such quick responses. The Schwann cell contains the typical cell organelles and cell membrane structure. However, notice as the Schwann cell surrounds the axon that the nucleus and other organelles are squeezed to the outside wrapping of the cell. This outer wrapping of the Schwann cell is called the neurolemma. The inner lining is made up of layers upon layers of cell membrane. This inner wrapping is called the myelin sheath. You will recall that the cell membrane, called the fluid mosaic model, is made up of a bilayer of lipids integrated with proteins. The thicker the myelin, in other words, the more layers of cell membrane making up the myelin, the more advantageous it is to the axon. One advantage is the regeneration of severed axons. Another advantage is an increase in the speed of the propagation of the action potential along the axon. The rest of this presentation will concentrate on the increased speed of action potentials down the length of the myelinated axon.
In fact, I can just jump to this part right here, and it will show you a comparison of two neurons. One of them up top here has the Schwann cells. It has multiple layers of insulation here. And then the neuron beneath it is going to be one of these unmyelinated neurons. So the axon has no myelination.
Compare the unmyelinated.
Here it is. And you'll see how much slower it takes this unmyelinated neuron. So this entire neuron could be existing within gray matter because it has no insulation whatsoever. Just the dendrites in the cell body here of this myelinated neuron, these could be in gray matter, but as soon as you see the myelination, you know this is part of the white matter axon portions that are myelinated. When there's myelination there, what you're going to see is that the electrical exchange, the sodium ions that move in and potassium ions that move out, they can only move in and out between those Schwann cells in these little gaps that we call nodes of Ranvier. Let me use another one of these weird analogies. Let's let the surface of the floor here represent the axon. And then maybe the bottom of my shoes are going to be the voltage gates that have to open and close. If this floor right here is the unmyelinated axon down here at the bottom, then if I'm going to get to the other side of the room, if I'm going to get to that axon terminal over there, then I've got to make sure that, you know, my feet are touching the floor. You know, I have to make sure that there's no gaps in this signal It's going to take a long time, but I'm eventually going to get to the other side of this room. I'm going to hit that axon terminal. So it's much slower if there's no myelination. This electrical current has to hit every part of that axon before it reaches this axon terminal, as opposed to the neuron on top, where there are these big Schwann cells. The Neuron Exchange, which I was representing by my feet hitting the ground, if there's Schwann cells that I have to step over, then I can get from one side of the room to the other side of the room much more quickly because my feet only have to touch on those nodes of RAND-VA, those little spaces in between. Let's see that with an animation.
Axon with the myelinated axon.
I'll back it up.
Compare the unmyelinated axon with the myelinated axon. You can see that action potential reached the end of the myelinated axon more rapidly than the unmyelinated axon. The speed of the propagation is faster going from node to node than action potentials that develop adjacent to the previous action potential.
Okay, fine.
So is that saying that, go ahead. And it's saying that white matter processes quicker because it has the myelinated portion?
We can say that action potentials will travel faster along the axon portion, right? Because information processing is going to involve the dendrites and the cell body and the axon. And if we're thinking about action potentials, we're just thinking about the axon portions. So the white matter is going to be myelinated axons. So action potentials could occur where there's white matter. But if we're talking about gray matter, it's any, cell bodies, dendrites, and even axons that are unmyelinated. So you could have slow action potentials taking place in the gray matter, or very fast action potentials taking place in the white matter. What were you thinking, Chanel? It's embedded in the PowerPoints. That link, when you click on it, you can see the whole video. Also, that email that I sent out at like 1 o'clock this morning, you've got pieces of this video kind of clipped into that one too. I made a separate little PowerPoint slide that is kind of got videos embedded in it. I thought it was just an additional helpful resource when we get into this stuff. Here's what we're about to talk about next. Let's just watch this video as opposed to me drawing it on the board first. But what we're about to see is exactly what I was trying to draw, where there's one neuron that's talking to another neuron. And as we're seeing the first part of this video, it's going to look exactly like the neuromuscular junction stuff we talked about last time. But instead of these neurotransmitters hitting a motor end plate on a muscle cell, they're going to be hitting receptors on the dendrites of another neuron. And I think I just added this video link to the PowerPoint slides, and I did update all of this last night, maybe at midnight. So if you haven't checked it since midnight last night, it should be updated. But here's this one.
A chemical synapse is a special junction between two neurons that allows them to communicate without being physically connected.
Can you check if we're good?
A synapse has several structures. The presynaptic neuron ends in a small bulb called the presynaptic terminal. The postsynaptic terminal membrane is directly adjacent, separated by a small space called the synaptic cleft.
Sucks. No.
An action potential causes voltage
I haven't seen anything for Temple
calcium ion
College,
channels
so
to open in the presynaptic
I wouldn't go to your
terminal.
ACC classes
The
today.
influx of calcium ions prompts synaptic vesicles to release neurotransmitters via exocytosis. This neuron is releasing acetylcholine. The neurotransmitter quickly diffuses across the synaptic cleft. Two acetylcholine molecules bind with one ligand-gated sodium channel at the postsynaptic membrane, opening the channel. When enough sodium ion channels open, the postsynaptic cell depolarizes and the action potential continues along the neuron.
I'm going to back that part up just one.
Open. The postsynaptic cell. When enough sodium ion channels open, the postsynaptic.
So that little thing, it said when enough of those sodium ion channels open, then that postsynaptic neuron sends its action potential. The thing that I was drawing on the board last time when we were talking about that neuromuscular junction, there was one of those slides of that neuromuscular junction where the motor neuron was talking, instead of talking to a dendrite of another neuron, it was talking to that motor end plate. And we said if enough neurotransmitter hit that motor end plate, kind of like those little rocks that were thrown onto the surface of the water, if the disturbance was great enough, then it would cause that motor end plate to to hit those voltage-gated channels on the sarcolemma of the muscle. And that's kind of what's going on here. There's no motor end plate, there's no sarcolemma, but we've got these chemically gated channels, those same non-specific ion channels located on the dendrites. When acetylcholine hits these channels again, they open up, they let sodium come in, a little bit of potassium goes out, but more sodium comes in than potassium goes out. So there's a swell in positive charges, and when enough positive charges make their way into this cell, we can see it fire one of those action potentials. So whether or not one of these cells sends an action potential has something to do with the amount of sodium ions that move into the cell. If enough sodium ions move in, it's going to send one of these signals. We'll just watch it go to the end there.
Depolarizes, and the action potential continues along the neuron.
Okay. 2 quick videos. This, just to show you where this is at, if you go to D2L, find our class on the D2L page. You have to click under the course home tab, but it will take you to this link that I put up. If you click on this link, it will bring you to this page, which is the same thing that we are about to talk about. And I just wanted you to have recorded versions of me going through these things so that you can watch it over and over. You have to go click on View the Slideshow, and it pops up like this, where as you click through,
the thin extension
videos are already loaded properly. You can just kind of watch it.
These dendrites receive signals.
I think this is where I got some feedback that sometimes the videos, I guess if you don't go to the next slide, they take you down a YouTube rabbit hole or something. You're watching a French bulldog on a motorcycle or something.
As the activated red signals dominate, an electric pulse is sent through the thick lower tube of the neuron.
Anyway, let's see if I can, I don't know if I could stop that, but what that slide was showing basically was just that the neurons are always sending signals from the dendrites towards the axon terminal. They're like one-way streets, which again is what we're seeing there. We're seeing the signal go from the axon hillock down towards that axon terminal. So each neuron is like a one-way street. Let's see what else.
Membrane potential.
I'll pause this, but we're about to talk about how these cells are capable of generating electrical currents. It's going to take us into talking about membrane potential. I just want to show you that there are videos here that you can watch. We'll talk about the sodium-potassium pump.
Okay, when we're considering
and then you even have the stuff that I'm about to talk about here, and the graphs that we draw locked and loaded. Let's see if I can get out of this.
this time,
Okay, so trying something new with the technology. But I'm going to get out of our neuron review PowerPoint. I'm going to pull up the neurophysiology PowerPoint because we've set some of this stuff up already. I don't know how far I'm going to be able to get through all of this stuff. I will say at least that today's lab is extremely short. It's just looking at these two models, so I may have to use some of today's lab time to get a little bit ahead in lecture. But before I press any further into lecture, if anybody wants to take a 5-minute break or so, stretch your legs, do whatever you need to do, feel free to do that. On the other side of this, we'll just jump into the neurophys PowerPoint. Okay, we're going to do this a brand new way. I'm going to try and leave the chain of neurons that are talking to one another on the top part of this board up there. And we're going to use the space on the bottom half of this board so we can always look up and kind of reference what it is that we're graphing here. I want to do a couple of things, I guess, with these diagrams before we get into the slides. And it's really just kind of drawing these neurons and talking about how they have a charge, initially a negative charge, and how they're capable of sending these electrical currents from one cell to the next. And I've already got a few of these either chemically gated channels or voltage gated channels drawn on here. I just thought we could make a list of the types of channels that we're going to see in operation when we talk about neurons. The first type of channel that I don't have up here at all, these are called leak channels. Leak channels are always open. They're like a straw or a hollow tube. They are specific to things. So there are leak channels just for sodium ions. There are leak channels just for potassium ions. There are leak channels just for calcium ions. But leak channels are always open, as opposed to the other types of channels that we're going to look at that are gated channels. Let me grab my colors for gated channels. We said there are chemically gated channels. They open and close based on the presence of a chemical signal. Sometimes these are called ligand gated. The ligand is the chemical signal that docks to the channel and causes it to change shape. So ligand gated channels and chemically gated channels are the same thing. And then we've got our voltage gated channels. Again, there's a gate there, but this gate opens and closes based on a voltage. We'll say voltage gated sodium ion channel. We'll have our calcium voltage gated channel. And the last one we'll put up is our voltage gated potassium channel. Okay, so we've got leak channels, we've got gated channels, and in any channel, whether it's a always open channel or a gated channel, when these channels open, they're going to allow ions to go from an area of high concentration towards an area of low concentration. Does anybody have any perfume or body spray or anything like that? Front row? What is that?
Like a oil that you roll, but it smells good. I don't know.
Well, so these molecules are in very high concentration inside this bottle, and if we, I don't know if this is strong enough, but if we were to open it up, and I won't waste it and let it sit there the whole time, but the smell should slowly permeate from an area of higher concentration out towards an area of lower concentration. This type of movement is just due to concentration gradient. Things spread from low to high things spread from an area of high concentration out towards an area of low concentration. So concentration gradient is one way in which things move. I'll let that be sufficient for that. Might not have got a chance to get all the way across the room. So like I was saying, channels allow things to go from an area of high concentration out towards an area of low concentration. But there's one more thing we need to add to this, and it's instead of a channel, it's called a pump. And unlike a channel, a pump is going to actively use energy to push things against the concentration gradient. It's like swimming upstream or against the current. You got to invest a lot of energy to do this. So one of these pumps that we're going to mention is the sodium-potassium pump. So it's the only one of these channels to actively use ATP. We got a list of channels there. Let's finally get to drawing our neuron. I'm going to, I guess, just use this space on the part of the board here to draw our plasma membrane. Of a typical neuron. And neurons, if we were to look at this plasma membrane on a typical neuron, well, let's add, we'll see the first thing that we can add to this is the fact that neurons are going to have a bunch of these sodium-potassium pumps all along their surface. And we're going to see these sodium-potassium pumps work in detail. What they do is they're going to use ATP. Maybe I'll just show an ATP molecule going up to this pump and then being broken down into an ADP plus an extra phosphate ion. And as this pump is basically breaking down ATP, what it's doing as it breaks down ATP, we're going to see that it's going to throw to the outside of this cell 3 sodium ions. And at the same time it throws out 3 sodium ions, it's going to pull in 2 of these potassium ions. And that's going on each one of these sodium-potassium pumps. So you can imagine 3 sodium ions being thrown out and 2 potassium ions being thrown in. And again, I'll just draw this on 2 of them to not crowd the drawing. But you can imagine after these sodium-potassium pumps work for just a little bit, we're going to see that inside this cell, we're going to have a very high concentration of these potassium ions. Any potassium ion that's floating around outside is likely to get thrown into this cell. So high potassium ion concentration on the inside and very low potassium concentrations on the outside. Compared to the sodium ions, any sodium ion that happened to be in the cytoplasm of this cell, which since we have our plasma membrane listed, we can maybe also put that inside the cell, this is our intracellular fluid, or the cytoplasm of the cell. To the outside of the cell is our extracellular fluid. That cytoplasm, or that intracellular fluid in neurons, has a very high concentration of potassium inside and a very low concentration of sodium ions. There may be a couple in there, but they don't stay there very long, because as the sodium-potassium pump works, it's going to throw those sodium ions outside and pull potassium ions back inside. So let me catch up with all of these sodium ions that we find in this extracellular fluid. Okay, that's enough. High concentrations of sodium in the extracellular fluid, low concentrations of sodium in the intracellular fluid. Contrast that with potassium. High levels of potassium inside that cytoplasm of the cell, low concentrations of potassium outside. Right now, we haven't added any of these voltage-gated sodium channels. But you can imagine if we had voltage-gated sodium channels along here, typically these voltage-gated channels are closed. But as we will learn, if this cell happens to depolarize past a point that these voltage-gated channels happen to open, you can see sodium is already stacked up on the outside of the cell. It would very quickly rush into this cell and cause the cell to take on a bunch of these positive charges. This sodium ion rushing into the cell is going to be what we call depolarization. If I can barely fit that in there. Depolarization, the cell is gaining these positive charges. Sodium ions are rushing into the cell. Now, we're going to see that there's really 2 reasons that sodium ion wants to rush into the cell. First off, the concentration gradient is such that there's so much sodium outside, very little sodium inside, that it follows that concentration gradient inside. The other thing that allows sodium ions to rush into this cell so quickly is the fact that inside the cell, inside neurons, you're going to have these negatively charged proteins. I'm going to draw them in these weird shapes. They just kind of fit in between the spaces of this cell. And these are big proteins that are carrying around a bunch of negative charges. So we're going to refer to these things as anions. And they're only located inside the cell. They're too big to fit through the plasma membrane and find them out here in the extracellular fluid. So when we look at those negatively charged proteins, remember opposite charges are attracted to one another. So the positively charged sodium is drawn towards the negatively charged anions. Sodium can rush into these cells very quickly. That's going to be what we call depolarization. Now, if we want to do the opposite, if we want to get rid of a bunch of positive charges, it would be difficult to shed a bunch of sodium because there's so much sodium outside the cell. The easiest thing to do to repolarize a cell is instead of getting rid of sodium, we can see that potassium is already set up to where there's a bunch of potassium inside the cell and very little potassium outside of the cell. So we're also going to see these voltage-gated potassium channels that are, again, specific just to the potassium ion. When these voltage-gated potassium channels open up, we're going to see potassium ions are going to leave the cell. And as the cell is getting rid of all of these positive charges, it's purging itself of these positive charges, and it's leaving behind cytoplasm with these negatively charged proteins. So when the cell is getting rid of these positively charged ions, this is what we call repolarization. So the cell getting rid of these potassium ions is what we call repolarization. I want to talk about how these voltage-gated channels for potassium and for sodium work. But at least we can see they are involved in either depolarization or repolarization. And the point about repolarization that I kind of wanted to make here is that it's a little bit more difficult for potassium to leave the cell than it is for sodium to rush in. Remember, sodium was rushing in because it was going down its concentration gradient and it was going towards the electrical gradient of these negatively charged proteins. Potassium, it's the opposite. Potassium is going to leave the cell because it's going to go down its concentration gradient. It's going to go from an area of high concentration out towards an area of low concentration, but it's going against the electrical gradient. These are positive charges, and they also like those negatively charged anions. So it reluctantly leaves those negative charges. So repolarization, like we said, is a little bit trickier than depolarization just because the concentration gradient is the only thing that's pushing these potassium ions out. The electrical gradient kind of wants to hold on to them. But okay, so we can see these negatively charged proteins are located inside of neurons. And that is why if we were to touch one of these neurons with like a voltmeter, we would see that the inside voltage of a neuron is typically about negative 70 millivolts. So one of the things we're trying to describe here is what's called membrane potential. Why are there a slightly different charge inside the plasma membrane versus outside the plasma membrane of a neuron? There's a slight negative charge, negative 70, to the inside of the cell, as opposed to the outside of the cell where there's a bunch of these positively charged sodium ions. In fact, this charge, this negative 70 millivolts, is going to be what we describe as membrane potential. I'll just write it up here as membrane potential is this charge that you find on the plasma membrane of neurons. And we described that it's got a negative charge because there are all these negatively charged proteins in the cytoplasm of that cell. And what we're about to graph here, when we graph how these neurons send these rapid electrical signals, it's going to have to do with, again, voltage-gated sodium channels opening up, allowing sodium to come into the cell. The cytoplasm then is becoming more and more positively charged as positive charges flood that cytoplasm. The opposite of that is our repolarization, where the cytoplasm is purging itself of positive charges. So let's actually try and graph this cell becoming more positively charged as it takes on sodium, and then becoming more negatively charged as it gets rid of potassium. You can stop me at any point if I'm stirring up questions. And I'm going to get just those names out of the way to give me a little bit more space.
So depolarization is when the cell is more positive than negative?
We would say depolarization is when the cell is becoming more positively charged. And let's graph that. Just try and use this thing. In this graph, we're going to put the charge of that cell. If we were to hit this plasma membrane with a volt reader, it's currently reading negative 70 millivolts. We're going to write the charge of that cell on our vertical axis. Membrane potential. We know this membrane potential is measured in millivolts. And we're going to compare that to time, which when we're talking about neurons sending signals, it's a very rapid signal. So we're on the order of milliseconds. This whole change in charge is going to happen within just a few milliseconds. So 1, 2, 3, 4, 5. Membrane potential. Let's kind of label this axis somewhere in this. I'll just maybe put it right in here. We'll say this is negative 55 millivolts. And I'll put a dotted line that's going to represent threshold. And just below that, we're going to get more negatively charged. So I'll put those colors down and grab this one. At about negative 70 millivolts, we're going to see this is when a neuron is just at rest. So kind of just like we've drawn over there, this is our resting membrane potential. A neuron is at about negative 70 millivolts due to all those negatively charged anions that are inside the cell. If we were to go down beneath this resting membrane potential, this takes us down into like negative 80. If we were to go the opposite direction, we would be going up towards the positive side of the scale. Let's maybe make this negative 30. Maybe make this 0 right in the middle, and up top we can go up to, say, plus 30. So maybe another way of showing what get that out of the way. Another way of showing what I'm trying to graph over here, if we think about the membrane potential of a cell when it's here at negative 70, like what we've just drawn over there, the intracellular fluid has a slight negative charge compared to the outside. This extracellular fluid, due to all of those sodium ions around the outside, has a positive charge. And what's going to happen in this span of just a few milliseconds, the cytoplasm of these neurons is going to go from negatively charged all the way up to, on this end of the spectrum, our intracellular fluid is going to become positively charged very briefly, and that negative charge is going to move to the outside. And then once the cell comes back down below this threshold, the cytoplasm inside is once again negatively charged. So we start down here at the bottom where we've got negatively charged cytoplasm. And the first thing that happens, if sodium ions were to rush into this cell, we know they each carry a positive charge. So if this line started to creep up like this, what that's showing us is that some sodium ions have moved into the cell. The cytoplasm has gone from negative 70 up to like negative 60. It's gaining some positive charges. And if it doesn't gain enough of those positive charges that we cross threshold, remember something magic happens right when we cross threshold, and that is it's going to involve voltage-gated channels. If we stay below threshold, we do not involve voltage-gated channels. These are all, below threshold is all chemically gated channels. So we're going to talk about sometimes these neurotransmitters are not in sufficient levels that this slight depolarization reaches threshold. In the video, there's another graph of an action potential that shows some of these slight depolarizations that fail to reach threshold. And if we don't reach threshold, then this axon hillock where the voltage-gated channels are located, like what I'm trying to put together here is this threshold, that little voltage-gated asterisk that I'm putting there, that is located right here on the axon hillock. So each of these, this is the first domino that has to fall if we're going to generate one of these action potentials. So if we're going to generate an action potential, it's going to start right here above threshold, and it's going to involve voltage-gated channels. Right now we have not involved voltage-gated channels because we have not yet crossed threshold. Okay, said that a couple of times. Now let's draw a signal that is going to depolarize all the way up to threshold. Let me know if I'm generating any questions that I'm just not looking at yet. It's okay, I'll put it right back up. well, I'll do it this way. That thing that we just saw where there was a slight depolarization, if we did not cross threshold, I'll grab the red color that represents my sodium-potassium pump. So this means that there has been a few sodium ions move into the cell. That's this little slight depolarization. And if we don't reach threshold, what happens is this sodium-potassium pump is going to work to bring us right back down to resting membrane potential. So here we are right back at rest. That's why it's sometimes described that one of the actions of this sodium-potassium pump, what we can say that it does is it helps maintain resting membrane potential. It helps maintain that resting membrane potential because if we get too far above or too far below that resting membrane potential, in this case we've taken on too many sodium ions, but we know if that sodium-potassium pump works for just a little bit, it's going to throw out those sodium ions and pull in potassium ions. So again, too many sodium ions into the cell, those sodium ions get thrown out, potassium ions get pulled in, and that brings us right back down to our negative 70 millivolts. I'm going to have to erase. Let's do that. Let's do exactly that. So if it doesn't cross threshold, sodium-potassium pumps just bring us right back down to where we typically started, this resting membrane potential. But here is where a depolarization has taken us past threshold. So I'm going to go ahead and put our little asterisk right here that says our first domino has now fallen, and all of these voltage gates are going to go through their series of positions. But let's graph this out real quick. Right after we cross threshold, this thing, we can graph it going all the way up to about plus 30, at which point we reach the peak of the action potential, and the charge of the cell has now taken on so many sodium ions that the inside cytoplasm is positively charged. But then this is quickly going to fall from plus 30 all the way down below resting membrane potential. I'm going to go all the way down to like negative 80. And then, to be specific, from about negative 80 up to about negative 70, this is our sodium-potassium pump. We said if we get too far below or too far above resting membrane potential, that sodium-potassium pump keeps us right back where we started, at that resting membrane potential. Now let's graph the phases that we talked about. As soon as we cross threshold, this whole part of the phase where the cell is becoming more and more positively charged on the inside, we said this was depolarization. This is due to sodium ions moving into the cell. Let's say influx. And the reason sodium ions are able to move into the cell is because those voltage-gated, I'll just say voltage-gated sodium channels are open. Since those sodium channels are open, sodium rushes into the cell, and the cell becomes more and more positively charged. But something happens right here at the peak of the action potential. At the peak of the action potential, we're going to see those sodium gates are going to close, and instead, these potassium gates are finally going to open. When potassium gates finally open, like we were seeing, potassium is ready to leave that cell. It's going to go down its concentration gradient, and as the cell purges itself of those positive charges, it's leaving behind more negatively charged cytoplasm. So this is what we call repolarization. It's due to those potassium gates being open, and when those potassium gates are open, potassium leaves the cell. So it moves out of the cell.
And at the peak, you said that the sodium channels close?
Yes. So repolarization is when the cell is becoming more negatively charged. But then look at this spot right here where we actually drop below resting membrane potential. This part of the graph where we are more negative than we normally are is called hyperpolarization. hyperpolarization is actually due to potassium ions moving out of the cell. And the reason these potassium ions are still moving out of the cell here is because these potassium gates are slow to close. Slow to close. We're almost there. This is about to make a little bit more sense. Any questions so far? All I'm going to do, I'm going to erase this thing because I want to draw one more thing on it. Okay, no problem. I'm erasing this, but this is basically simplified over here where we're showing the cell being more negatively charged when it's at resting membrane potential. The cytoplasm is more negatively charged compared to the outside. Then the peak of the action potential is when the cytoplasm becomes positively charged due to all of those sodium ions that have rushed in. And so that's the peak of the action potential. We've got sodium ions, we've got potassium ions in here, so it's really positively charged. But then during the repolarization, potassium ions are leaving, and as the cell's purging itself of those positive charges, it causes the cytoplasm to once again go back down to that negative end of the range. So this last bit, I'm just going to fit it in two different places. Stretch it out wide instead of stacking this on top of one another like I did in the video. Let's talk about the position of these voltage-gated channels. I'll do this another way. I'm going to use this space because it might be a little bit more roomy to draw the action of our voltage-gated sodium channel. So let's say voltage-gated sodium channels. When we draw these voltage-gated sodium channels and how they behave, I'm going to draw them just like this, like a series of boxes. And just to clarify what we're looking at here, this is the plasma membrane of a neuron, just like the one that I had erased that was studded with these voltage-gated sodium channels. This is our plasma membrane. So the fluid inside the cell is our cytoplasm. And that fluid outside the cell is our extracellular fluid. Okay. There are three different positions of this voltage-gated sodium channel, and here are those positions. I'll keep it like that. The first one, I will, on this side of the first half of this channel, I'm just going to put a small eye right there, and I'm going to put a circle around it. This little ball that's basically dangling from a thread there is going to be what we call the inactivation gate. That's why I put the I there, because it stands for inactivation gate. And on the side of this channel that faces the extracellular fluid, there's another gate that I'm going to label with an A. I'll put a little circle around that A, and I'll put the hinge attached to the other side of this protein. This, I'll just give myself a little bit of room here to label. I'm going to scoot my cytoplasm over just a little bit. So this first position of our voltage-gated sodium channel that I'm trying to draw, you can see there's no ions that can cross this membrane because the gate is closed. I want to say closed but capable of opening. It's closed, but it's capable of opening. It's closed, but it's active, is another way of describing that. As opposed to this next position. This next position we're going to see is going to be a position where this inactivation gate is open like it was originally. And the activation gate is also going to be very quickly flung open. In this position, this is the second position of our voltage-gated sodium channel. We call this the open position. It opens very quickly. And as this gate opens very quickly, what happens is, right behind that, sodium ions move into the cell. So we have sodium ion influx. Sodiums very quickly move into the cell. So many sodium ions are going to rush into this cell, we're going to see that it becomes very quickly positively charged on the inside. Let's just go ahead and add those on there, since I'm talking about them. This is our closed but capable of opening position. This is the position of all of those voltage-gated sodium channels when our cell was at normal resting membrane potential. Normal resting membrane potential, we said, was negative 70 millivolts. So at negative 70 millivolts, these neurons are at rest. Voltage-gated channels are closed, but they're capable of opening. What takes us from this closed and capable of opening position to being actually open is crossing that threshold value. All you have to do is go from negative 70 to negative 55 millivolts. If this cell goes from negative 70 to negative 55 millivolts, what we said crossing threshold, receiving a threshold stimulus is what flings open those voltage-gated sodium channels. So now sodium gates are opened. As soon as we cross threshold, sodium ions rush into the cell. The cell's becoming more and more positively charged up until we hit the peak of that action potential. So the third position here, if I can get the right color, our third position of this voltage-gated sodium channel is going to be closed and inactive. So it's closed and it's not capable of opening. What closes here is this inactivation gate closes off. The activation gate is still open, but the inactivation gate is closed. So we've basically closed this channel. Ions can no longer move through this. What allows us to close this channel is when we hit the peak of that action potential, plus 30 millivolts. When our cell hits this plus 30 millivolts, those sodium gates close very quickly. And so now sodium can no longer move into the cell. This gate is now closed off. The thing that I really want to draw, if I can put an asterisk on this somewhere, position 1 and position 3 are both closed, but they are not the same position. Position A is closed, but it's capable of opening as long as we hit threshold. This position number 3 is closed, and it will not open until it is reset back to this original position. So the thing, and let me get my purple color here, the thing that takes us from closed and inactive, this peak of the action potential, and resets us back to this original closed but activated position is when the cell repolarizes down past 70 millivolts. This repolarization down to 70 millivolts is what resets. It resets these voltage-gated channels. Let's go back over here and see what we're talking about. So resting membrane potential, we got voltage-gated channels that are closed. But then as soon as we cross this negative 55 millivolts, that's our threshold value that very quickly causes those voltage-gated sodium channels to open. As soon as those voltage-gated sodium channels are open, we can see sodium rushes into the cell very quickly. And then as soon as we hit plus 30 millivolts, this is the peak of that action potential, where those voltage-gated sodium channels close. So instantly, we no longer have sodium rushing into the cell. Instead, we're going to see potassium is going to start to leave the cell. Like we said, this voltage-gated sodium channel is going to stay closed until we repolarize down to negative 70 millivolts. I'm going to put a little asterisk there that represents negative 70 millivolts. This one represents negative 55. So we have our trigger to open the voltage-gated channel and our trigger to reset that voltage-gated channel. So 3 positions for the sodium-gated channel. I'm going to use just this little space right here to write the activity of that voltage-gated potassium channel. There's only 2 positions there, so it's a little bit easier. So voltage-gated potassium channel. Let's put our, try that again. Let's put our plasma membrane there with these gates in it. So this is our plasma membrane. Outside of that, we have our extracellular fluid. Inside of that, we have our cytoplasm. And we've got our voltage-gated potassium channels here. These have only 2 positions. So in this first position, we're going to call this closed. In the closed position, I'm just going to put this little circle attached to a little hinge that has closed this gate off. So potassium ions can't cross this membrane. The gate is closed. The only other position we're going to see is the second position that we call the open position. Again, this gate just is going to open up, and when it's in the open position, potassium ions are going to be able to cross this membrane. We know they're going to be moving out of the cell. And to get our triggers, we know what took this voltage-gated channel from the closed position to the open position. It's the same story. At resting membrane potential, all of these voltage gates are closed, but they're ready to spring into action. All we have to do is go from negative 70 millivolts and reach a threshold stimulus. If we cross negative 55 millivolts, that threshold stimulus takes us from this resting membrane potential all the way across threshold. That signal of crossing negative 55 millivolts is the signal that causes these potassium gates to open up as well. But I'll add this. So I'll say potassium gates open slowly. As opposed to those voltage-gated sodium channels that open very quickly, these voltage-gated potassium channels open very slowly. So they're actually told to open. That crossing negative 55 millivolts, that's the signal for those voltage-gated potassium channels to open. But you'll notice that there's a few milliseconds that pass before potassium ever starts to leave the cell. This is showing you that the voltage-gated potassium channels are, I think of them as like those apartment gates that are slow to open and slow to close. Have you ever gone to those apartments where there's a gate, you got to punch in a code, and then after you punch in the code, it takes like a couple of seconds for that gate to open up wide enough that you can get your car through. So that lag time where you're waiting for that gate to open up after it's already been told to open up, you see that with those apartment gates. And just like those apartment gates, they're slow to open, but they're also slow to close. And you see this in the apartments too. When the gate starts to close, it's still wide enough that some cars can fit through. So you'll have cars like haul ass through there real quick before the gate gets closed all the way. The same thing happens down here with potassium ions. These voltage-gated potassium channels are slow to open, and the signal that gets them to reset, how we go from open all the way back down to closed again, is by reaching negative 70 millivolts. These neurons have to once again repolarize all the way down to negative 70 millivolts in order to reset these voltage-gated channels. So let's look at it from the potassium's point of view. Potassium gates are told to open as soon as we cross threshold, but they're slow to open. So there's a little bit of time where there's just sodium that's moving in and no potassium is moving out. That's our depolarization. Then at the peak of the action potential, this is when simultaneously those voltage-gated sodium channels close and the voltage-gated potassium channels are opened wide enough that we now get ions that leave. Ions continue to leave that cell through those open voltage-gated potassium channels. Ions are going out until they repolarize all the way down to negative 70 millivolts. Once we repolarize down to negative 70 millivolts, that's the signal for these voltage-gated potassium channels to close. But again, they close slowly. And what happens as they are slow to close, just like at those apartment complex gates where people rush through those gates before they actually close, some of these potassium ions are leaking out of the cell because those gates are slow to close. The loss of excess potassium ions leaves behind even more negatively charged cytoplasm. This is what causes us to go below negative resting membrane potential for just a few milliseconds. And the sodium-potassium pump, once again, takes us back up to resting membrane potential. It throws out the excess potassium and pulls in some sodium ions. Yeah?
So if we go back to the beginning, ATP is what causes those sodium-potassium pumps to open?
You went back to sodium-potassium pump. Okay, so if we're thinking of ATP, then ATP is basically going to be used to establish this concentration gradient of a bunch of sodium outside the cell and a bunch of potassium inside the cell. Remember, the sodium-potassium pump works by creating this concentration gradient so that there's way more sodium outside the cell than there is inside the cell. And when these voltage-gated sodium channels finally open, sodium is going to move into the cell because there's very little of it inside the cell. Yes?
So the first one we drew, the voltage-gated NA channel, that's when the cell is depolarizing, and then to get it back to repolarization, the calcium channels have to open? Is that?
There's no calcium channels here. The only voltage
Potassium.
That's right. So for the cell to repolarize, voltage-gated potassium channels are going to have to open, and potassium is going to have to leave the cell. And when enough potassium leaves the cell, the more potassium that leaves, the more negatively charged we become. And we have to be able to get rid of enough potassium to get us all the way back down to that negative 70 millivolts. And that's what resets all of those voltage-gated channels.
My question was, what causes those sodium channels to want to open? Like, what is that threshold? What gets it to the negative 55 that causes them to want to open?
What is an example of an excitatory signal that can get us? So my example, my hypothetical example, was us standing on the dock throwing little pebbles into the surface of the water. And if it's just a slight disturbance, then that ripple is never going to get up to threshold. But if it's a big enough rock where the disturbance is sufficient enough that it's going to cross threshold, then that's going to shake open our voltage gates. And the example that I might have used over here, when I'm just tapping on my leg like this, I can feel that on the surface, like the surface of my skin. I can feel that I'm touching something there. But this is not a sufficient stimulus to get my muscle to contract. And if I were to hit it hard enough, somewhere in there, without thinking about it, I should be able to get this muscle to contract. There's a little bit of one. So sometimes the stimulus isn't sufficient enough to cross threshold. Or like... Well, reflex is an example of that. That's the muscle responding to a stimulus. And I think your question is the stimulus, whatever the stimulus is, maybe it's cold, maybe it's, in my case, touch, maybe it's sound. If the initial stimulus isn't sufficient enough to cross threshold, then your neurons won't process it. Like sounds, for example, if it's a very faint sound, you might not be able to pick it up because your neurons aren't sensing that. But if the call from the bird or whatever is loud enough, then it might cross threshold and your neurons are going to pick that up.
So kind of what we're talking about right now, does this fall into so we get a tap on our skin and we hear a sound, that's like our sensory neurons, and then this is like the integration and motor neurons, like processing and then creating this function, like the signal?
It is tough when we learn about all of those in one day. But it would be the case that the sensory neuron is sending an action potential to an integration neuron. And that integration neuron, so in the three neurons, let's say, I wish we could say this is a sensory neuron, but I drew it as a multipolar neuron. And I erased the other sensory neurons. Sensory neurons are going to have dendritic ends that are feeling around for some type of receptor. Maybe it's sound, maybe it's cold, maybe it's touch. Once they're triggered to cross threshold, then the dendritic end of that sensory neuron is going to send an action potential to a motor neuron.
And the threshold that starts it is the axon hill block.
Yes, great question. Here's what I need to do, and I think I did this in the video. So this part of the graph, with at the risk of getting this thing even more confusing. This whole part of the graph below threshold is when we're dealing with graded potentials. And then as soon as we cross threshold, this whole part from, well, I'll use maybe this color, I guess. So from here, once we get above threshold all the way down to crossing this again and hyperpolarizing, the whole second half of this is the action potential. So remember, action potentials are as soon as we cross threshold and are involving voltage-gated channels. Before we cross negative 55 millivolts, these voltage-gated channels aren't doing anything. Remember, the voltage-gated channels here are the first domino that we need to get to fall if we're going to get this action potential to go all the way down the axon to the axon terminal.
So we have to pass the 55 before it starts.
That's right. So this thing, the axon hillock, is the negative 55 threshold thing. Like this is the first domino that we have to get to fall. In order to get that first domino to fall, we have to get this axon hillock to depolarize to negative 55 millivolts, because that's going to set into motion all of these voltage-gated channels doing their things they need to do to generate this action potential. Prior to that, this is when we're standing on the dock throwing those little pebbles into the water. And some of those rocks might cause a disturbance that's big enough to generate an action potential if it triggers those voltage gates. But if the little pebbles are not causing a big enough disturbance, then it's never going to involve voltage-gated channels. And that's where that first disturbance that we were drawing, that if it's only a slight disturbance, then that sodium-potassium pump is going to bring us right back down to resting membrane potential. So the initial stimulus has to be what we call sufficient enough to cross threshold. What was that?
So basically, if you take a boulder.
Yes, that's right.
To rattle everything to be able to depolarize it so that it can do what it does to make it come back down to the
Right, and I'm just sitting here tapping on my knee, but I use that example. You can actually put those electrical stim machines onto somebody's muscle, and you can dial up how much of an electrical signal you want to hit that muscle with. And if you hit it with just a slight stimulus, it's not enough to get a contraction. That's not a threshold stimulus. But if you dial it up strong enough to cross threshold, then you're going to get that muscle to contract, or the neuron to fire, whatever it may be.
Okay, sorry for the question, but just to clarify, so when we are in between the resting membrane potential and the threshold, it's graded potential. Then everything above that is the action potential, and once we get back to threshold, it's back to graded potential.
Yeah, so anywhere on this graph where we are involving the activity of those voltage gates, that's going to be what we consider the action potential. And this little part here, but between when the neuron is at rest and when it's sending an action potential, this is the area that we called graded potentials. And graded potentials, if we can go back to map what we're talking about here, I'll come to this little section. Let's say we've got a neuron here that's just at rest. We can use either one of these, I guess. We've got a neuron here that's at rest. It's not doing anything. All these voltage-gated channels are closed. In order for this neuron to generate an action potential, we know that action potential starts right here. Right? It starts as soon as we involve voltage-gated channels at this axon hillock. So everything before that that's going to involve chemically gated channels, cell bodies, and dendrites, this is all below threshold. So this is the graded potential area. If there are enough neurotransmitters, like we said, if the boulder is big enough, then there's going to be enough sodium ions that rush into this cell body that get a depolarization of negative 55 all the way over to that action, all the way over to the axon hillock, which generates that action potential. Like we said, if the neurotransmitters are not sufficient enough, that there isn't enough sodium ions that rush in, that slight disturbance kind of fizzles out before it hits that action potential. If we only get to negative 50 here, those voltage-gated channels never open, and this signal of neurons communicating comes to a stop right here. And we want to know how is it that the signals jump from one neuron to the next. So we have to get this axon hillock to depolarize past threshold.
So like if someone's paralyzed and they like can't move a leg, their like axon potential is not going above threshold, so their signal doesn't go all the way to their legs.
That's right. There's something that's preventing, that could either be sensory, they're not feeling something, or if it's motor, then they can feel it, but they're just not able to get that to move. So that's right. So those action potentials, they happen in sensory neurons going back towards the brain and spinal cord, or they can go in the opposite direction away from the brain and spinal cord to the motor neurons. And that's why it was so helpful, I think, to have these neurons in here so that you can kind of see it's action potentials followed by these graded potentials. And if the graded potential stays sufficient enough, then we will generate another action potential that will lead to another graded potential that may or may not stay sufficient enough to generate another action potential.
So then what you're saying is that right there, so the RMP right there is the graded potential, and then over there, right, that's.
So this is a neuron at rest, it's not doing anything yet.
Right.
But now there's been a disturbance to this neuron.
Right.
It may or may not be sufficient to cross threshold, and if it does cross threshold, now we're going to generate an action potential because we're getting into that first voltage-gated domino falling.
And then when you get down to where it says negative 70 on the purple side, is that considered graded potential as well? Or, yeah.
Here's what that's called. I'm trying to figure out a color that I haven't used already, and maybe this light blue one will work. So let me do it like this. I'm gonna put a dotted line, a vertical dotted line that just goes right through this first threshold point of negative 55. And I'm gonna do another one that goes right through that threshold, the second asterisk value here of negative 70. So before we hit this first dotted line, we have not involved any of those voltage-gated channels. They're all sitting at their ready-to-go position. They're closed, but they're activated. And as soon as we cross threshold, we said this is the area in which those voltage-gated channels, like the sodium channel is open. After the peak, that sodium channel is going to be closed, but it's not yet reset until we cross this negative 70 millivolts. So this space in between here is what we call an absolute refractory period. An absolute refractory period is a time period when you cannot generate a second action potential until these voltage gates reset. So I think, was it Colleen or somebody just had a comment about neurons not being able to send another signal. And one thing that can stop neurons from being able to send a second signal is if these voltage gated channels can't reset. So in order for us to get out of this absolute refractory period into this little segment here where we are below resting membrane potential, but our voltage gates have actually reset. So they're back to this ready to go position, but we're further away from threshold. See how much further away from threshold we are than we would be at rest. So this part of the graph here is what we call the relative refractory period. Relative refractory period. If I can spell it, refractory period. This relative refractory period is a time where it's not an absolute refractory period. You could generate another action potential in this little time, but you would need a stronger stimulus than a normal threshold stimulus because you're further away from threshold than you normally would be at rest. So absolute refractory period is this time where the voltage gates have opened. They're still inactive. They can't generate a second action potential until they reset all the way back to the beginning. Here's another thing that is, I don't know, maybe worth, it might help with the understanding. If you have ever experienced hyperkalemia, or anybody that's ever been hyperkalemic? Is this the question? Nope. Hyperkalemia is a situation where you have too many potassium ions outside of the cell. I'm just going to use this neuron over here. What we said, typically neurons have a very high concentration of potassium inside, and a very low concentration of potassium outside. But in individuals that are hyperkalemic, they have too many potassium ions in the extracellular fluid. Hyperkalemia is excess potassium ions in the extracellular fluid. So imagine somebody that's hyperkalemic like this. I know I've erased the picture, but we said when voltage-gated potassium channels open, potassium wants to leave the cell and go out into the extracellular fluid because typically there's more of it inside the cell than there is outside the cell, except in individuals that are hyperkalemic. You can imagine if there is more potassium outside of the cell than there is inside the cell, when those voltage-gated potassium channels open, instead of potassium leaving the cell, potassium may go back into the cell. If anything, it's going to be reluctant to leave. And here's the thing that's fatal about hyperkalemia. If you are hyperkalemic to the point where neurons cannot repolarize past negative 70 millivolts, if you can't get enough potassium ions out of your cell to repolarize down to negative 70, maybe you repolarize down to negative 69. It's not down to 70 yet, so those voltage gates have not reset. You're locked in this absolute refractory period. Your neurons can't send any more signals. That leads to organ communication failure, and that's usually fatal. I can pull down the slides and we can try and catch up with some of those slides, but I feel like the key slides that we're going to see going through there are the things that we diagrammed. I don't know how I'm doing on time here, but one of the things that I wanted to explain is the phases of an action potential, that you can recognize depolarization from repolarization, that you know whether, what ions are moving during depolarization. It's sodium ions, and they're moving into the cell. And then what ions are moving during repolarization, and what way are they moving? It's potassium ions, and they're moving out of the cell. And then same thing for hyperpolarization. What ions are moving during hyperpolarization, and what direction are they moving? Once again, it's potassium ions, and they're moving out of the cell. So knowing the phases, the positions of those channels, and whether they're opened or closed would be important. Knowing what the difference between absolute refractory period and relative refractory period is not as important, but I'm sure that's going to be something that's mentioned on the exam. And again, absolute refractory period is a time where we have started those voltage-gated channels in motion, but we have not yet reset them again. So they can't generate another action potential until they have been reset, which is that negative 70 millivolt point. Again, after they've reset, you have now, they're ready to go again, but for a fraction of a time period, you're hyperpolarized. So the strength of stimulus that it's going to take to reach threshold is going to be about double what it normally would be when the neuron's just at rest. I want to go backwards and just show a few of these slides just so that you can click through this for a quick refresher. It's kind of everything that we mentioned today. So here is the short version of the slides that I've posted in the Google Slides.
The thin extensions at the top of the neuron.
I like this one because it shows the direction of that action potential. I'm going to kill that light for a second.
As long as the inhibiting blue signals dominate.
You can barely see it, but there's that electrical impulse that we are basically trying to graph. This wave of sodium coming in and potassium going out. They're showing as the little electrical color. Just distinguishing dendrites from axon terminals. And then you can see a bunch of different types of neurons all sending signals from the cell body towards the axon terminals, kind of indicating that they're all one-way streets. And this is just showing the difference between myelinated axons, where the signal goes from the axon hillock to the axon terminals faster than in unmyelinated axons. So we saw that. Now here's the thing that we were just talking about with membrane potential. Let's see if this 2-minute clip is better.
Or membrane voltage refers to the difference of electric charges across a cell membrane. Most cells have a negative transmembrane potential. Because membrane potential is defined relative to the exterior of the cell, the negative sign means the cell has more negative charges on the inside. There are 2 basic rules governing the movement of ions. They move from higher to lower concentration, just like any other molecules. Being charge-bearing particles, ions also move away from like charges and toward opposite charges. In the case of the cell membrane, there is a third factor that controls ion movement. the permeability of the membrane to different ions. Permeability is achieved by opening or closing passageways for specific ions, called ion channels. Permeability can change when the cell adopts a different physiological state.
Yep. Here's that sodium-potassium pump.
Active transport is a type of cell transport that requires the input of energy in the form of ATP. The proteins that
This one's
conduct
quieter,
this form of transport are often called pumps because they force molecules or ions to move from an area of low concentration to an area of high concentration. This is commonly referred to as against the concentration gradient. One of the more common examples of active transport is the sodium-potassium pump. The job of the sodium-potassium pump is to move sodium ions, or Na+, out of the cell, and potassium ions, or K+, into the cell. Let's take a closer look at how this happens. The sodium-potassium pump has binding sites for 3 sodium and 2 potassium ions. Let's follow the movement of the sodium ions first. After 3 sodium ions are positioned within the carrier protein, an ATP molecule is split, releasing phosphate. This phosphate binds to a location on the exterior of the carrier protein, causing the protein to change shape. As the protein changes shape, the 3 sodium ions are released to the other side of the membrane. Next, 2 potassium ions position themselves within the carrier protein, causing it to undergo another change in shape. In the process, the phosphate molecule is released. Once the phosphate molecule is released, the carrier protein expels the potassium ions into the interior of the cell. The carrier then resumes its initial shape, completing the cycle. Notice that for every 3 sodium ions leaving the cell, 2 potassium ions enter. Since both sodium and potassium ions have a positive charge, this unequal movement causes an electrical gradient to develop across the plasma membrane of the cell. A number of cellular processes, including the generation of nerve impulses, use this electrical gradient.
Right, so there's more sodium ions outside the cell than there are potassium ions inside. And also inside the cell, there are all those negatively charged proteins. So that's also what contributes to that negative charge of the cytoplasm inside the cell. And here's another little thing that you can kind of see with the pictures. There's 2 positions that this sodium-potassium pump can take. It's either open to the cytoplasm of the cell or it's open to the extracellular fluid. And look at when it's open to the cytoplasm of the cell, those little docking sites for sodium are just in the right shape that sodium can nestle its way in there. And then look what happens when it opens to the outside. In this other shape, those little docking sites for sodium, they change shape to where sodium doesn't quite fit in there like it used to. And that's what allows sodium to go ahead and break away from this and drift out to the extracellular fluid. You'll notice in the open to the outside position, these binding sites for potassium are now just in the right position for potassium to dock. When before, when this was open to the inside, those docking sites for potassium were not quite in the right shape for potassium to dock. So in this position, we would say it has a higher affinity for sodium ions. Sodium loads up, it gets a phosphate and changes shape, and now that it's changed shape, sodium can no longer dock there quite as easily, but instead potassium is now ready to dock in this position. But then once it loads up with potassium, it loses that phosphate. It goes back to its original open to the cytoplasm position where potassium ions can't really stay there. But the docking sites for sodium are now back to their normal position, and sodium is going to get loaded up. So sodium-potassium pump is constantly working to throw sodium outside and potassium inside the cell.
Potential.
The same thing that we just graphed, you've got a slower version of this on here.
Let's
and
define this
what I like about this is that you've also got the, even though it's cut off from this overhead projector, you've got the graph of an action potential. This were those stimuluses that failed to reach threshold. So instead of ever affecting those voltage-gated channels, that sodium-potassium pump brought us right back down to resting membrane potential.
Charge is about 100 millivolts worth of a change in the charge.
So anyway,
that by doing a
I'll click through this. You can see we got the same thing that we just listed. I was trying to talk about absolute and relative refractory period, and that's why I put this last slide on here where I tried to use these different colors to indicate this first box is our absolute refractory period. It starts as soon as we cross threshold, as soon as we take those voltage-gated channels from their closed and not do anything position to their open position. They've crossed threshold, so that first domino has fallen. We don't reset those voltage-gated channels until we once again drop back to negative 70 millivolts, which is obviously cut off from this screen. You'll have to take my word for it. But as soon as we cross negative 70 millivolts, that resets all of those voltage-gated channels. So we're out of our absolute refractory period and into this relative refractory period, where voltage gates have reset, but we're further away from threshold than we normally would have been at resting state. So it needs a stronger than normal stimulus to get us up to threshold. But this diagram, all I do here is graph the positions of those voltage gates that we talked about. And it should cut off at a certain time, but I have noticed that sometimes the edits that I make don't stick, and they change and sometimes may lead you down a rabbit hole of other videos, which is not intentional. So I'll continue to tweak that if possible, but the unedited, unchopped up video of this is already up on D2L for you to watch the same thing that we just did today over and over, or you can watch it through these Google Slides. That was the last one. I know I didn't get to pull up any of these neurophysiology slides, but that's where we can start next time with, once again, a review of this physiology stuff. 2 lectures on this, and then that'll give us 1 day to point out brain and spinal cord model stuff. This is really the part of the unit number 4 where the physiology comes in, just talking about how these neurons communicate to one another, how they send signals. So I think we've gotten all the way through our class handouts for over here. I'll stop talking about new stuff, let you take a break if you want, since we still have about 30 minutes left of lab time. If you want to stick around, I'll go through these models of the 2 neurons. It's just the front page of the lab list. I'll just pull up a chair and sit right down here in front and point out both of these the neuron models, and then just make laps and answer questions.